यहाँ से दिखेगा नहीं ना, क्लैरिटी भी आएगी।
हाँ सर,
सर आप कर दो स्टार्ट।
बिल्कुल आएगा।
ऑन है सर ये।
हाँ ऑन है।
हाँ ये इधर, मैं इधर घुसती जा रही हूँ, ये कैमरा इधर।
हाँ मैं सेट करता हूँ, इधर आ रही हूँ।
नहीं मैं
आप आ जाओ ना, मैं बोल रहा हूँ तो पोस्ट मैं बंद कर देता हूँ। Welcome to all for this e-poster session. We are sharing e-posters of all the participants one by one. Participants are requested to unmute themselves and to describe the poster. The time allotted to each participant is 3 minutes. So this is poster number 1 of Suchitra Sharma. Ma'am, please unmute yourself. Ma'am Suchitra Sharma, are you there?
ये नंबर वाला है ना सर, यही है।
नीट है।
मैसेज किए हुए सर।
फुल स्क्रीन कर, फुल स्क्रीन आप कर रहे हो
Presentation by the participants who sent their e-posters displaying on the screen. Every participant has given 3 minutes to display his work and poster. And judges are Dr. Dipti Srivastava Madam, Dr. Anami Barlow sir, Dr. Arun Chakravarti are visualizing the e-poster. Award will be given on valedictory function. Poster is rolling on the screen. Students of MSc, please visualize the posters. You may also take inspiration from these posters. It is not very hard to prepare posters and presented them in various international conferences. Only 3 minutes will be given to the participants. Please summarize early.
मैं ऑडिबल हूँ ना, म्यूट है इससे। ऑडिबल है। हेलो,
Yeah, you are audible.
am I audible?
Yes.
Ma'am, this is not my poster.
Okay, what's your name, please?
Suchitra.
Yes,
Yes,
please.
ma'am. Yes, ma'am. Can I start?
Yes, sure.
Okay, so I would like to start with the introduction. As we all know, radiation exposure causes oxidative damage to macromolecules, which leads to oxidative stress, inflammation, and apoptosis, eventually causing tissue damage. So this raises the need for development of safe and efficient radioprotectors. Q3R is a flavonoid which is found in a variety of plants. It has antioxidant, inflammatory, and radiomodulatory properties. It has been used in the clinics to treat heart ailments, but studies regarding its radioprotection mechanism and gastroprotective role is limited. My objectives are to evaluate the potential of Q3R to elevate radiation-induced oxidative stress, inflammation prevention in the irradiated intestine, as well as to study the regulation of NRF2 and NF-kappa B signaling pathway.
That's all. Have you finished?
No, ma'am, just 2 minutes. My methodology includes selection of optimum dose of Q3R in A549, selection of NRF2 inhibition dose of ml385 in A549 by RT-PCR and flow cytometry, then study of NRF2 expression levels at mRNA and protein level in different experimental groups, and study of protein expression in irradiated GI tissue. So, based on the cell viability test, we observed that Q3R does not cause any alteration in cell viability up to 72 hours, and Q3R improved cell viability in A549 cell post radiation treatment. Can you please scroll it?
Scroll up.
No. No, ma'am.
Somebody, you want to say somebody in conclusion?
Ma'am, can you please up it a bit from the title?
Right.
Yes. Then we by ML385 inhibition studies, we observed that Q3R exerts radioprotection by upregulation of NRF2 in A549 cells, and in radiation group, NRF2 expression was decreased, whereas in Q3R group, NRF2 was increased, whereas when inhibitor was used, it was decreased. Then in mice model, we use 7.5 gray of radiation dose, and it was observed that pre-treatment with Q3R protected the mice intestine against radiation-induced oxidative injury, as evident in the figure. Q3R plus radiation group showed significant improved antioxidant as well as reduced glutathione levels as compared to the radiation group, which was otherwise reduced when inhibitor was used. Now to conclude, I would say that Q3R exerts radioprotection by upregulation of NRF2 as well as downstream proteins and downregulation of NF-kappa B and other inflammatory mediators. Summary and conclusion is that Q3R confers radioprotection by modulating NRF2 and NF-kappa B crosstalk pathway. My future plan is to study the additional signaling pathways of radioprotection which may crosstalk with this pathway. I would like to acknowledge DRDO, Director in Mass, CSIR for Fellowship, HOD, Officer in Charge of Animal Facility and Radiation Facility. Thank you.
Next poster is Lipsy Goel, please sum up within 3 minutes.
हुआ नहीं है अभी.
Am I audible?
अक्षु दैया है.
Yeah, actually, I'm actually right now. Am I audible enough?
Yes, yes, please.
But my poster is not displaying there. It's Lipsy World's poster. Like, I can't see my poster on the screen.
Lipsy Goyal, please.
No, actually, it's Akshudaya. She has to open her poster. Yeah. Yeah. Well, I'll start now.
Yes.
Good afternoon, everyone. My name is Akshu. Currently, I'm working as a research scholar in INMAS. You have zoomed it. Well. Yeah, it's okay. The thing that I'm working on is GIRS, that is gastrointestinal acute radiation syndrome. Whenever anybody is exposed to total body radiation, it is quite difficult to study specifically GIRS due to bone marrow failure. So I am focusing on GIRS study and effect of my drug, that is trichostatin A, as a radio mitigator to study its effect on the GI syndrome. Can you scroll it a bit down? Yeah. Trichostatin A, the molecule that I have used is, it's a pan-histone acetylase inhibitor. Its role has been known for majorly for myelosuppression. So its mechanism of action have not yet been studied fully, and it is currently in clinical trials against cancer treatment. Scroll it down. Yeah. My major objective of this study was to study the effect of TSA on acute GIRS. Majorly, firstly, I have acclimatized my C57 male mice 4 days earlier before the study. Then the mice were, I think it's fine. Then the mice, my abdomen was localized with 15 gray abdominal irradiation, where my head, thorax, and limbs were scheduled with 5 centimeter lead, which provides 95% shielding to the mice. And then TSA were administered 1 hour 24 hours post radiation. And then the study lasts up to 10 days, where I have carried out majorly the molecular studies and GI studies, which were majorly observed at GI damage is observed after 10 days of the radiation. My methodology basically includes basic study, that was body weight, stool output, diarrheal index, and I have carried out the structural integrity studies using HND and PES staining, and metagenomic studies using 16S rRNA sequencing. The molecular studies majorly involves my immunoblotting, ELISA, the immunohistochemistry studies. It's a bit the basic results. First results that I have studied were basic health parameters. These were body weight loss, as you can see from the graph, that the body weight loss of the 15 gray mice up to 10 days severely hold from 1 to 10 days, while TSA treatment also showed some weight loss, but that was significantly improved as compared to my 15 gray groups. Similarly, the number of fecal pellets per mice were also observed daily up to 10 days. A major difference was found on day 7 post-radiation, where the fecal count was severely reduced post 7 days. The diarrheal index, the diarrhea has been observed after my radiation treatment from the day 4, and it lasts up to 8 days in my 15 gray group, and TSS treatment as usually have showed some improvement. And disease activity index, it has been calculated based on my body weight loss, number of fecal pellets, and blood in the stool of the mice. Since I haven't observed the blood in my stool of the mice, so this factor was not considered. So this the calculation of this activity index was majorly involves the body weight loss and number of fecal pellets. Can you move it up a bit? Then I'll move to my next result, which are the HND stain. Yeah. Can you move it up a bit? More slightly up. Yeah, that's it. Then I have studied the effect on the GI morphology using hematocillin and eosin staining, where I have found reduced number of crypts and villi post radiation. While surprisingly, there was no effect on my villi length. And the PES staining, which majorly shows the PES staining, which shows number of goblet cells, will also reduce post radiation. The microbiota studies, majorly we have calculated the number of total fecal bacterial species.
Conclude, please.
So I have observed that the TSA have do have a potential to protect my GI against GI, acute GI error. And my future objective is to set the delayed effect of TS, like delayed effect of the GI error using TS as a residue mitigator. I would finally, I want to acknowledge my DRDO for my funding, Director Ilmas, my HOD, Professor in charge, and the lab member.
Thank you.
Thank you.
Next one is Lipsy Goyal. Please sum up within 3 minutes.
Good afternoon, everyone. As I am audible?
Yes, yes, please.
Yeah. So this is Lipsy Goyal, this side. Here to present my work having the title Protective Efficacy on N-acetylcysteine against the combined radiation and burn injury in a mice model. So briefly introducing my work, that's combined injury is a syndrome that can be considered as a thermal trauma that is associated with the radiation injury. Will you please scroll it down a bit? Yeah. So I choose my drug, that's N-acetylcysteine. It's an antioxidant and usually work as a mucolytic drug. Now please scroll it down to methodology. So methodology involves an oral instigation of NAC drug prior to radiation within a 0 minute. The C57BL6 was chosen made mice for a 30-day model observation. After radiation of 5 gray, after 2 hours, whole body burn all about 85 degrees for 10 seconds was given. And then the basic parameters, enzymatic assays involving antioxidant assay and oxidative stress measurements had been done. So ma'am, please scroll it up to the up results. Yeah, that's ma'am, just skip the introduction part. Yeah, thank you. So as the burn is the one which I am being described, that's wound healing has been delayed due to radiation exposure. So the quantification of wound size and the scar fall had been considered. These images from different days had been taken. That's a comparison of a burn wound involving the combined wound. Combined wound involves the radiation exposure over it. And then I had checked that combined wound when NAC was installed in that mice. So at different days, stay. Okay, thank you. Just stay. So the wound at day 6, day 9, day 13. Till day 13, the burn wound, the alone burn wound is all about to cure, while the combined one is almost that's the size has not been decreased yet, and NAC was up to an improved version. So till day 17, the scar had already been formed of burn wound as well as of NAC, but the combined was not yet to cure. So at day 20, we can easily see the NAC group. NAC group, the scar has been fall down, but at day 26, the combined wound had shown that it had been cured. So coming on to the quantification, scar fall, combined wound had taken much time to cure rather than NAC is working well.
Lipsy, please conclude it.
Yes, please scroll it down. Then the gross, just stay, sir. Just yeah, thank you. So gross parameters, the body weight change, food percentage, food intake, and water intake had been checked out, and it had been shown that it's decreased significantly after the combined radiation wound when given, and NAC had shown much improvement in that. So please scroll it down. Furthermore, antioxidant status and oxidative stress had been checked, in which NAC had worked successfully. So my future work, this model had been worked for 30 days. So next, in a way forward to work for NAC at different time points. So observations was that the NAC had worked well in this. So thanking Director in Mass, HOD of MARV, and Dr. Pawan, Dr. Anant, and Dr. Ravi for the Animal House facility and DRDO funding. Thank you so much.
Thank you. Next one is Pratibha Kumari.
Yeah, am I audible?
Yes, Pratibha Kumari, please sum up within allotted time.
Okay, thank you so much, sir. So, I'm Pratibha, and I'm here to present my poster on amelioration of radiation-induced gastrointestinal injury by radioprotective drug RKIP-006. So, in a brief introduction, gastrointestinal system is the second most radiosensitive biological system, and gamma radiation induces massive damage to intestinal crypt stem cells, which lead to GI impairment. Right? And so further, my aims and objectives are evaluation of radioprotective effects of bacterial molecule RKIP-006 against gamma radiation-induced gastrointestinal damage. My experimental designs are to assess the radioprotective efficacy of RKIP-006, which have the parameters which are studied. First, morphological alterations. Second, cell viability. Third, cell proliferation. Fourth, DNA damage. And fifth, oxidative stress. So can you, yeah, thank you. So the methodology for the study is divided into the 4 groups: control, radiation plus drug, and drug only. And we take IC6 cells for the in vitro study model, which is derived from the rat small intestine and which also shows the characteristics of the crypt stem cells. So here, after IC6 were pre-treated with drug before radiation exposure, we studied the 4 parameters: morphological analysis, in which cells were observed under microscope to determine morphological alteration, and the cell survival through MTP and clonogenic assays for the cell proliferation and metabolic activities. Third, the DNA damage using Comet assay, and fourth, loss determination using fluorescent dye BCFDA. So, yeah. Yeah. So here are the results for the study. The first, morphological analysis. As you can see here, I have chosen the 2 time points, 24 hours and 48 hours. In 24 hours, there are less differentiation amongst the radiation and the radiation plus drug treatment group. But we can see little damage as control to the control group, right? But in the 48 hours group, as we can clearly see that in the radiation group, there are large number of dead cells, and the cell to cell attachment is also lost, right? Yeah. So yes, school side. Yeah, thank you. So the second result is radioprotective efficacy evaluation in intestinal IEC6s through metabolic activities and cell survival. So in metabolic activity, as we can see here through MTP assay, there's a significant difference between the radiation group and the radiation plus drug treatment group. Yeah? And in the second cell proliferation survival assay, as you can see here, there is also significant changes between the radiation and the treatment group. So the third results are DNA damage using Comet assay. In this assay, we can see a comet in the form of DNA, damaged DNA, which is in which electrophoresis is used. So here we can see that the tail DNA percentage, which shows the damage in the cells, is higher in the radiation group and lower in the treatment group. And we can also see here the percentage olive tail movement is same, showing same significant results. Yeah. Yeah. So the last result is oxidative stress evaluated using flow cytometry. We can see here the microscopic analysis. We use the DCFDA dye, which is fluorescent dye, and here we can see that in radiation group, it shows
Please conclude it, please.
Yeah, it shows high intensity of dye, which conclude in the form that more damaged cells take the more dye and show more intensity fluorescence. Yeah. So in conclusion, RKIP6, which are drug, protects against the radiation-induced damage in intestinal cells, and significant time-dependent increase in DNA damage also observed, and ROS level significantly decline in the drug-pretreated group. And my future plans is radiation-induced inflammation and its modulation will be evaluated in IAC6 cells. And I'm here to acknowledge INMAS DRDO and DBT for providing research facilities and funding. Thank you so much.
Am I audible, sir?
Yes, yes, please.
Thank you, sir.
Pushpa, please conclude within 3 minutes.
Okay, ma'am. I, Pushpa Meena from Department of Zoology, University of Rajasthan. Our topic of presentation is application of metabolomics in prediction of lymph node metastasis in head and neck cancer. Head and neck cancer is 6th most common malignancy that affect more than 500,000 people worldwide each year. Screening of this cancer is continuously improving, but still the mortality rate is high. The prominent reason for this is lymph node metastasis. The capacity to prevent this mortality largely depend on the early diagnosis of metastasis. The collective finding indicate that oncogenic metabolism can regulate metastasis. For this, here the comes the role of metabolomics. Metabolomics is an emerging field of system biology which quantitatively measure the small molecule metabolites in body fluid like serum, saliva, and urine, and tissue also by analytical techniques like LCMS, GCMS, and NMR. Metabolomics study target on altered metabolic pathway to find specific biomarker for diagnosis and early prediction of disease. Methodology. For this study, we collected serum sample from of the patient from Bhagwan Mahavir Cancer Hospital, Jaipur, which was approved by the ethical committee of the hospital. We collected serum sample of healthy control head and neck cancer patient and without lymph node metastasis. We collected each number of sample was 20, and the prepared sample were analyzed by using LCMS system, which was coupled with ThermoQ executive plus operating in positive and negative ionization mode, and the LCMS have C18 column, and the result generated raw file by LCMS were analyzed with compound discoverer software. Metabolite annotation was performed through ChemSpider and MZ Cloud database. ChemSpider database search was performed with BioCYC and HMDB and KEGG database. The obtained data was analyzed using the one-way ANOVA. Here I shown my result of ANOVA by heat map. Here we can see the elevated level of many some metabolites like amino acid, fatty acid, and choline containing phospholipid compounds occur in the group. Conclusion. This study provides some altered metabolite in head and neck cancer lymph node metastasis, which can be used as metabolite biomarker. These biomarker can be helpful as diagnostic marker and potential therapeutic targets also. In the final, I would like to acknowledge my Department of Geology and Mahavir Cancer Hospital for providing me the facility. Thank you.
Thank you. Last one is there.
So me, Akanksha.
Yes, Akanksha, your poster we are sharing within a minute.
Okay, sir. So can I share my poster?
Yes, please share.
Okay,
Yes,
sir.
Akanksha, please share.
Yes, sir. So is my screen visible?
Yes, Akanksha, please go ahead.
Good afternoon to all. My name is Akanksha Bhardwaj, and thank you for giving me the opportunity to present this poster. I'll be presenting on radioecology, dissecting complexities of radionuclide transfer under climate change. The radioecology deals with the distribution and transfer of radioactive substances in the environment, and also the risk it is posed to the man in the environment. The radionuclide is the ubiquitous part of our life, and there's no way to escape from them. And they are included with the immense application in the military, medicinal, and industrial sector. Among them, the major peaceful application of radionuclide is in the nuclear sector by owing the 16% of the world electricity. In further addition to challenges caused by the climate change, transforming the non-motile form of the radionuclide to the motile ones and causing the contamination to the soil and water bodies, also risking the contamination in the environment as well. The radionuclide is in the food and environment is a serious concern. These imaging are depicting the real-time image of the iron in the barley, and it is clearly indicated that the shoot is absorbing more amount of energy, and also the transfer from root to shoot in the iron deficient plant of barley is more if we compare it to the iron sufficient one. These Poisson emission tomographic image is also showing the carbon transport in the life plant, that is tomato. In conclusion, or the take-home message is that the radionuclide is having immense application, and their decay will emit the energy which can be used for the agriculture sector, such as cereals, fruits, and vegetables can be used for the prolonged period by gently radiating them. Seeds can also having increased yield by radiating them, and also we can check the measurement of the fertilizers by the radiation. Thank you.
Thank you, Akanksha.
Next session start. नेक्स्ट सेशन भी स्टार्ट करवा ये करके नेक्स्ट सेशन स्टार्ट करवा
Thank you for the nice presentation.
माइक ऑन कर
Thank you for the nice presentation. Now next section will be started.
तो करो स्टार्ट
Young scientist. Next, Young Scientist Award.
Very good afternoon. I'm very sorry if we are running at least 1 hour late. So, as we decided in the organizing committee, we will promote the young researcher. Just, I'm very happy to see in this audience, most of the audience they are our postgraduate students and research scholars. Dear students, Young Scientist Award may be given to 1, maybe given to 2, 3-year-old, but the participation in this award session is much more important. Those all are participating in this award session, I have our blessings for them. The member of our senior member of our ISRB organizing executive council, Professor P. Venkatachalam from Ramachandra University, Chennai. I have been twice to Ramachandra University. I have seen the laboratory of Dr. Venkatachalam. Are you here? Since
yes, sir. Good afternoon, sir.
Dr. Venkat, I am very sorry, Venki, for since now you have been waiting for me.
Not at all, sir. Not at all, sir. It is my pleasure to be part of the program.
Welcome, Dr. Venki. I have been to your laboratory.
Yes, sir. Thank you so much, sir. Thank you so much for the invitation.
Such a senior faculty member, great scientist, is our chairperson. I am very happy to see you with us. Professor Veena B. Kushwaha, Madam Veena, ma'am, are you online?
Yes, sir. Hello,
Yeah,
sir.
Madam, again, madam, I am very sorry and we are thankful to you. And I have requested you also.
Part of conference, sir. No
Yeah,
problem.
yeah, madam. Lot of works have to be done. Madam Vinay Kusawa, we have been together in many conferences in ISAR also, and she had been one of the main speaker in the interest webinar organized by the department. We have with us most active persons of our department and very energetic. Dr. Archana Purohit is here. Archana, she is also one of the invited speaker. Chetana Arora, Chetana ji, we are also thankful to you. You have come from this adverse situation from Surat, traveling a long distance. I would request all these chairpersons, there are 5 plus 1 more participant we have to add.
Dr. Tina Haritwal.
Dr. Tina Haritwal. One name was left. So there are 6 participants. I would request to take only 5 to 7 minutes to present the work. I know it is very difficult to present work in 5 to 6 minutes, very difficult. Because you are also a young scientist, but just we have to see how you present, what is your course content, and after 6 to 7 minutes, and the chairperson are requested to give us the sequence, like first number, second number, third number, fourth, and the 6th number. Okay? We don't want to discuss any participant. So I hand over the mic to Dr. Archana and Venkatachalam, and Adyaman is here. Adyaman, are you here? Aradhana Acharya, Aradhana, she's also here. Consider Tanmay Simali, okay. And the Pooja Rani, she's from, okay, you are here. And Anushka Sharma is online, and one more speaker, Tina, she's also online. So I hand over the mic, Professor Venki. Thank you, Venki. Welcome. Archana, you have the dais. Archana, Dr. Archana, both dais. Now I'm not going to, I don't want to stand in between the young scientist and the very senior scientist. Thank you so much.
Dr. Purohit, it is my humble request that the people who are sitting on dais should start the session so that online there may be some problem of interaction. You can please request Dr. Archana Purohit to start the session from our side. Thank you.
Good afternoon to all of you. I request to Dr. Archana, please start your.
The topic of my presentation is reduction of dose to the comparative best for patients undergoing radiotherapy by using super flat tissue material. As an introduction, as per Democon for the year of 2020, breast cancer is the most common cancer diagnosis and the leading cause of cancer-related deaths in the world. In females, breast, cervix, uterine, ovary, corpus, uterine, and lung constitute 50% of all cancers, as per the cancer statistics of 2020. And to deal with CA breast management, radiation therapy is an integral part along with the chemotherapy and surgery. So we cannot avoid radiation therapy. Though radiation therapy plays a vital role in local regional control, it is also associated with the delivery of scattered dose to the normal, such as present near to the tumor. So there are a lot of effects when we treat the patients with radiotherapy. These are the things I have highlighted. So there is the unfortunate thing with radiation is that it not only cures, it also induces the cancers. That is the main disadvantage of radiations, and we should use it properly. So there is a definite probability of second cancer in the contractile breast, which would be increased by any radiation dose received during the treatment. So the radiation dose received by the contractile breast during primary breast radiation is of very much concern to us when we perform the treatment planning, because low to moderate dose of radiation to the CB, that is the contractile breast, has been associated with the increasing risk of developing a second malignancy, and my study is in this region only. So this is the background. This curve shows that it's the most important somatic effect of low dose ionizing radiation. High frequency of breast cancer introduction has also been reported following exposure to radiations. So this figure shows the incidence of breast cancer as a function of dose for 4 different populations that allows risk estimates to be made. So this is the background. So as far as International Commission of Radiation Protection is concerned, in the year of 1991, the tissue weighting factor was given as 0.05, but in the 2007 they have increased the weighting factor of the breast, considering the issues associated with the radiation delivery. So literature review has been performed before conducting my study. In 1989 itself, people started investigating on this domain. They have measured the contractile breast dose during half beam block and isocentric treatment techniques for patients treated with primary breast. In 1992 also, Boyce et al. Conducted a study. They evaluated the risk of second cancer associated with radiation therapy to the breast. And in 1994, some study has been conducted to reduce the scatter dose to the contractile breast. In 1996 also, some study has been conducted. They have evaluated 4 different breast treatment techniques with 6 cm. In 2002, DL compared the breast cancer incidence rates after radiation exposure in 8 large cohorts. In 2004, Bhattacharya et al. Conducted some studies. Around 2007, our Professor Choge sir has also did some study in measuring the contractile breast dose. Now coming to my study, what is the aim? The aim of my study is estimation of total dose received by the contractile breast during radiotherapy, and also investigation of the role of superflag material of 1 cm thickness in modifying the dose to the contractile breast. So the materials and methods: 50 post-mastectomy carcinoma breast patients were included in the study. 50 gray was delivered into 1 fractions. These are the routine things we follow: conventional fractionation. The treatment was delivered in the linear accelerator machine. We have used calcium sulfate dysprosium TLD disc for the dose measurement. The dose was measured for each patient without the superflag for 10 fractions and with for another 10 fractions for subsequent comparison. This is the age distribution of patients and the state distribution of patients. This is how I have placed the TLD disc. The distribution of the TLD disc over contractile breast was such that it covers the complete surface area. So I have divided into a column. The medianmost column disc lie at a distance of 5 cm from the patient's midline. The TLD discs were left in place during the entire daily fraction of treatment, including the supraclavicular sheath. The same set of TLD was used for each patient. Here I have divided the breast into 4 quadrants, that is medial lateral, medial upper. So dose measurement procedure: in the first week of treatment, that is first 5 fractions, the patients were treated without any dose measurement or intervention for dose reduction, allowing the patient to settle. So in the next 10 fractions, patients were treated as regular with the placement of TLD disc over the contractile breast. In the remaining 10 fractions, patients were treated in a similar fashion as above with the placement of 10 mm superflag. So the TLD discs were processed and were read in the TLD reader available in the institute. So this is the linear accelerator in which I have treated these patients. And you can see the bonus material placed over the contractile breast. These are the materials I used for annealing the TLD discs, and that's the TLD reader. As for the results, the mean dose per fraction received by the contractile breast without superflag was 7.82 plus or minus 2.26, while the mean dose per fraction received with superflag was 3.78. So total dose received by CB breast without superflag was 191.14 plus or minus 65.62 centigrade, and with superflag was 94.69. These are the results. We can see that whenever the scatter radiation is following the inverse square law, when we move away from the contractile breast, the dose is decreasing. We can see that the red color is with superflag, the dose is reducing. when we are using a superflux, and the blue one is without superflux. So comparison of dose to the contracted breast with and without superflux. These are the results. We can see 48.57% reduction in the medial upper and medial lower, and lateral upper 49.51, lateral lower 49.87% are the reduction we are observing. So an average reduction in the mean dose in the presence of 1 centimeter thick superflux as compared to absence of superflux was 46.57 plus or minus 17.18%. So the reduction of dose on comparing two situations was approximately of the range of 20 to 80% and was statistically very much significant. So these are the bar graphs showing quadrant-wise dose distribution to the contractile breast. So as I mentioned earlier, we have divided the non-tumor breast, that is the contractile breast, into 4 quadrants. Also, we have taken the nipple part as a central quadrant. Medial upper, medial lower, central, lateral upper, and lateral lower. You can see that lateral lower is it will be the very much far from the primary field, and hence it is receiving the lesser dose. So discussion: the average total surface dose received by the contractile breast in the study was 191.14, which was 3.82% of the prescribed total dose. So the study conducted by the Chobley sir had shown a similar dose statistics, and the CB dose in the study was 152.5 to 254.75 centiliters. A similar total prescribed dose and dose fraction as in our study. A reduction in dose was observed on moving from medial to lateral quadrant, which I have explained just now. It's evident by the fact that the radiation reaching CB or scattered radiations are very less energy, and thus amount of these radiations decrease gradually on moving away from the radiation field. So Stowell et al. Also had a similar observation like this. So as a conclusion, using superflux is an effective method for reduction of dose to the contractile breast. It's an easy, convenient, and low time consuming method. Elicitation of any role in reduction of second malignancies in CLB now needs large studies with long follow-up. So these are the references I have used. I acknowledge also Dr. Purohit sir. Dr. Anantule is my guide and my HOD, and my senior professor, Dr. H2x Assay. These are the radiation equipments we have at our center. Thank you.
Thank you, Dr. Antraman. If anyone asks any questions? No question? The next speaker is
Excuse me, madam. Excuse me, can I interact with the presenter?
Yeah, sure.
Yeah, yes. It is a good presentation. There are two clarifications. Okay. So first one, you measured the dose to the tissue using a calcium sulfate as a TLD material, right?
Yes, sir.
The dose which you presented is an average to the different regions or any one particular region?
Yes. It is average, sir. Actually, we have taken 50 patients and we have taken the average of the 50 patients.
Okay.
It's not of one patient.
So what was the fold reduction in the total dose?
Pardon, sir?
What was the fold reduction in the total dose?
Sir, around 48% we are getting reduction, sir, overall, average.
Okay. So what is the translation potential for your study?
Sir, I'm not getting, sir.
No, you have conducted the study with the patients, right?
Yes, sir. Yes, sir.
So what would be the translation potential? Whether it can, the thing can be translated for everyday's treatment plan?
Yes, sir. We can incorporate also.
Or you need further studies to confirm your results?
No, sir. We have already implemented it. For the past 2 years, we are applying this bonus for every patient. This is very much conclusive study. For every patient, as a default, we are using bonus material, that is a superflux material over contractile breast for every breast patients undergoing radiotherapy in our department.
Okay, thank you.
Thank you.
Next, Ms. Aradhana Acharya from Basic PG College. please sum up within 5 or 6 minutes.
Yes, ma'am.
Good afternoon, everyone. Myself, Aradhana Acharya, from Department of Geology, Basic PG College, presenting my topic, the role of Ayurveda in cancer treatment. So, what is cancer? Disease in which some of the cells grow unregulated and uncontrollably and spread to the different parts of the body. It is a deadly disease all over the world. It may form tumor, it can be cancerous or non-cancerous. These cells can form anywhere in the body and increases up to trillions of cells. So, type of cancer, the cancer is divided into two types, benign cancer and malignant cancer. Benign cancer, in this, the cancer's growth is limited to a certain point, whereas in the malignant cancer, the cancer's growth spread to the nearby tissues of the body. On the basis of the position of cancer, it is divided into three types, that is to say epithelial, non-epithelial, and leukemia. Next, what are the symptoms and diagnosis of cancer? Symptoms depends on what part of the body is affected. General signs are as, but are not specific to cancer. Fatigue, lump under the skin, weight changes, skin changes, difficulty breathing, hoarseness, unexplained bleeding, etc. The diagnosis of cancer, the first one is the physical examination by feeling lumps or seeing the abnormalities like skin changes or enlargement of cancer. The second one is the laboratory exam by reading of blood test or by blood count test. The third one is the imaging exam. In this, we examine the bones and the internal organs of the body by computerized tomography, MRI, CT scan, PET, etc. The fourth one is a biopsy. In this, by collecting a sample of cells for testing. Next, what is the role of Ayurveda in cancer treatment? Ayurveda treatment focuses on maintaining health using Rasayan treatment. While treating the tumor cancer, it also helps in increasing the immunity of the patient with the help of meditation and yoga. There are several herbs which help in the cancer treatment. The first one is the guggul. It has the properties of painkiller, anti-tumor action, and inhibits abnormal cell growth. The second one is the kanchna. It has the anti-cancerous properties. It inhibits abnormal cell growth and help in bleeding disorder. The third one is guruchi. It's help in boosting immunity and reduce stress. The fourth one is triphala, that is to say amalki, vivithi, and harithi. It has anti-cancerous properties. It boosts immunity and reduces oxidative stress. The fifth one is trikatu, that is to say shunthi, pipli, and marich. It increases the digestion, increases the immunity, and has anti-inflammatory action. The last one is the Varuna. It has anti-inflammatory action and it cures limbs. The triphala in cancer treatment. Triphala means three fruits: Terminalia cabula, Terminalia ballerica, and Ambelica officinalis. They are present in the ratio of 1:2:4. It scavenges free radical, reduces oxidative stress, and non-enzyme level decreases lipid peroxidation. It reversed as chemopreventive, chemotherapeutic, and anti-cancerous properties. It modulates multiple all signaling pathways, including ARK, P53, cyclin G1, MAP, tubulin, etc. Triphala as chemopreventive. It has therapeutic and preventive efficiency against malignant cancers such as prostate, colon, breast, lymphatic cancer, etc. It reversed by in vivo studies. It has chemopreventive potential against cancer. In studies, reduces benzoatrine induced for stomach induced in stomach, papillomagenesis in mind, reduced tumor by 77.77%. It suppresses growth of capan to pancreatic tumor and xenograft reduction due to increased apoptosis in tumor cells by activate P53 and ARK. Triphala as antioxidant and pro-oxidant. It holds potential in restoring antioxidant level and decrease lipid peroxidation. This oxidant associated with the presence of polyphenols, flavonoids, and vitamin C. It quenches the ROS level, reducing reducing the oxidative stress. Pro-oxidant by inducing ROS production in cancer cell increases the level of the ROS production in cancer cell, causes lethality. It inhibits proliferation and induces apoptosis through ROS production, and it also produces radiosensitizing action through oxidative damage. Triphala has anti-cancerous properties. Triphala has shown anti-cancer activities in various cell lines. The aqueous extract of Triphala decreases the proliferation of breast and prostate cancer cells. It shows that the extract was rich in polyphenol gallic acid, which was considered as a major factor in inducing cytotoxicity of cancer cells. Increasing concentration of Triphala correspondingly decreases the viability of cancer cell, viability of treated breast cancer cell NCF7 cells, and induce apoptosis in NCL7 and PARC95 cells in vitro. With similar concentration of Triphala, it does not cause any cytotoxic effects or DNA damage on normal breast epithelial cells. NCF7 can have human peripheral cells, non-mouse liver and spleen cells. Triphala suppress cell migration of cancer cell in vitro, thus indicating anti-metastatic potential. Now I want to conclude my topic by saying that Triphala has been used for centuries against various elements in Indian traditional medicine system. Studies in the recent past have indicated that Triphala has immense potential in the reduction of oxidative stress or damage, as well in the prevention and treatment of the cancer. In different combination, ratio is required with the hope that new formulation may exhibit better beneficial effects on oxidative stress mediated chronic disease. Thank you so much.
Thank you, Aradhana. There is any query? Only one question allowed.
Can we have full form of these terms? Full form of the terms in particularly types of cancers.
Aradhana, one more question.
You are using Triphala.
Yes, sir.
Can you explain or can you think about that? Which chemicals that is useful for the process? Triphala itself has some chemicals.
Polyphenol gallic acid.
Which chemical is very useful for your work?
Doctor, what is happening in the medicinal sciences? The different scientists, they are using the different plant products. What he posed to us, what are the constituents of Triphala? You are not supposed to do the answer. What are the constituents of Triphala and what are the effect of each and every constituents? Since to MSc final, lot of work has to be done for the, like one of my students are working on the Amblica, like the aloe vera. Some of them they are working on the Moringa oleifera. They have not worked on the. I think our father is a great pharmacologist in Bikaner, Dr. R.P. Acharya. I think you are Dr. R.P. Acharya?
Yes.
So just he has used the crude acid only. What are the antioxidant activity? I think you have worked on oxidative stress, and the other protective efficacy of Triphala in the cancer. So how herbals are used for the cancer treatment? I think you have given emphasis on the general aspect of Triphala. Thank you so much.
Our next speaker is Tanmay Shrimali from Basic PG College.
Good morning, respected dignitaries. My name is Tanmay Shrimali. I am from Department of Geology, Basic PG College. Today I am going to present my topic on the correlation of ozone and UV rays. I am going to cover these following topics in this talk. first are the introduction. The atmosphere is divided into 4 layers, and the ozone layer is present in the troposphere and stratosphere, as we can precisely say that it is precisely present in the lower stratosphere. Ozone layer is a layer where the concentration of ozone is very high as compared to the other part of the smooth atmosphere. We can say that the concentration of ozone is 10 parts per million in the ozone layer, where as compared to the others it is 0.3 parts per million. Ozone holes are the phenomenon which are described as the ozone layer holes. We can say that ozone holes are the depletion of ozone holes. Ultraviolet rays. Ultraviolet rays are the rays which comes from the sun, and on the basis of penetration, it is divided into 3 parts. First are the UVA, UVC, and UVC rays. The ultraviolet rays I can start from this. I'm going to present my topic on the correlation of UV rays and ozone. The introduction. Ozone layer is the atmosphere is divided into four layers, and ozone layer is present in the troposphere and stratosphere. More we can say that precisely it is present in the lower stratosphere. Ozone layer is the layer which protects us from the harmful UV radiations which come from the sun, and it is the area where the concentration of ozone is very high. We can say that ozone layer is the area where the concentration of ozone is 10 parts per million, and other parts of the atmosphere where the concentration of ozone is 0.3 parts per million. Ozone holes are the phenomenon. Ozone holes are the phenomenon which are used as the area where the ozone layer is depleting, and next thing are the ultraviolet rays. Ultraviolet rays are the radiations which come from the sun. It is from the wavelength of 10 nanometer to 400 nanometer. The atmospheric nitrogen completely wiped out the general ultraviolet frequency, which is from 10 to 100 nanometer. On the base of the further penetration, it is divided into 3 parts: UVA, UVB, and UVC. UVC are the ultraviolet radiations which are completely wiped out by the ultraviolet rays and which are harmful to our skin. UVB are the wavelength which is from 315 to 218 nm, and these are the wavelengths which are halfway screened out by the ozone layer, and they reaches the earth's surface, and they are the main cause of the skin cancer and cataract. UVA are the radiation which are not completely wiped out by the ozone layer, and they reaches the earth's surface, affecting the life forms on it. Now we will discuss the stratospheric distribution of the ozone layer. The ozone layer are mainly formed over the tropics, and then they are transferred to the poles by the atmospheric wind patterns, which are described by the Brewer-Dobson method in the northern hemisphere of our study. And the main relation between ozone and ultraviolet layer are the ozone-oxygen cycle, which allows the formation of the ozone. In this first step, the dioxygen molecules is broken down into 2, and the broken oxygen molecules then collides with the other molecules of the oxygen and forms the ozone. This picture depicts the formation of ozone from the ultraviolet rays, and it is we can say that it increases the thermal energy in the stratosphere, making it warm, and it can be reacted with the other gases present in the those. Next phenomena, and which is also a social concern these days, are the ozone layer depletion. Ozone layer depletion is the gradual thinning of ozone layer by the presence of other harmful gases and increasing amount of them. The main cause of the ozone layer depletion is the ozone depleting substance, which are CFC, chlorofluorocarbon, bromide, halogen, and other. The ozone depleting substances are the substances which are the main responsible for the depletion of ozone layer and making the affecting the formation of ozone layer. It has the various effects on our environment. First, on our human health, the ultraviolet rays effect can cause the skin cancer and cataract, and it can also affect our immune system, can lead to it weakening of it. Next are the its responses on the animals, and in animals it basically affects their seeing ability and eye power. The marine life is also affected by it. Affects the formation of the plankton in this, which disturbs the food chain of the marine animals. Next thing are the effects of implant. It can implants the rays can affect the growth and flowering and photosynthetic process of it. Conclusion. The ozone layer is the layer which protects Earth from the harmful radiation. It is naturally formed in the atmosphere by the process of oxygen ozone cycle, and ultraviolet rays and UV rays. Ultraviolet rays and ozone are closely related with each other, as the formation and depletion both are interdependent on it, and ozone layer depletion is the major concern of the modern day world, and which we have to look after it. Thank you.
Questions? And then basic. Next, Pooja Ravi. Pooja, please complete within 5 or 6 minutes.
Hello, good evening, everyone. Myself, Pooja Khatri, Radiation Biology Laboratory, PG Department of Zoology, Dungar College, Bikaner. So my topic is enteroprotective action of Moringa oleifera against radiation and mercury alterations in musculars. Aims and objectives. Increasing use of ionizing radiation for diagnostic as well as therapeutic purpose has drawn attention of many radiobiologists toward the undesired side effects of these such exposures. Apart from ionizing radiations, human beings are continuously exposed to a wide range of metallic pollutants from the environment. Man releases lots of chemicals in the environment by mining, smelting, discharging industrial waste, agriculture and domestic waste, burning fossil fuel, and by use of pesticides. Mercury occurs naturally in the Earth's crust. It is released into the environment from the volcanic activity, weathering of rocks, in fertilizers, and as a result of human activity. Combined action of ionizing radiation and other agents are potentially great importance because there are many occasions where interactions might occur in our environment. Environmental contamination by mercury is a serious threat to all our living kinds. Modification of radiation and mercury chloride induced responses obtained by means of chemical substances that can significantly decrease the magnitude of response when present in biological system. This type of modification is classified as the chemical protection, and the substances responsible for it are called the chemical protectors. A large number of compounds have been investigated for the protective action by the different researchers, but these protectors are highly toxic at their effective dosage level except the MPG. The Moringa oleifera plant has been identified as an extremely useful medicinal plant, which is also known as the Sahajani in our Rajasthan, which is which is extremely used for medicinal plant with significant health benefits. Secondly, it is a claim to be completely non-toxic. Even at higher dose levels, it has been shown to act as a potent chemoprotector. Therefore, the present study was undertaken to evaluate the role of Moringa oleifera in modifying the radiation and mercury-induced changes in the jejunum of Swiss albino mice. So, materials and methods that have been used: animals 6 to 8 weeks old male Swiss albino mice were procured from Lala Lajpat Rai University of Veterinary and Animal Sciences, Hisar, and maintained at the 20 to 25 degrees Celsius. The animals were provided with the standard mice feed and tap water. Drug. The drug we have used, that is the Moringa oleifera in a powder form, which was procured from the Uma Lakshmi Organic Private Limited, Jodhpur, and aqueous extract of the same was obtained and in the department. The plant extract of the moringa was fed orally at the dose of 150 milligram per kilogram of the body weight. So the drug was given from the 7 days prior to irradiation or mercury chloride or the combined treatment. Source of irradiation: a cobalt gamma radiotherapy source is used to irradiate the animals. This facility was provided by the radiotherapy department of the Prince Vijay Singh Memorial Hospital, Bikaner, Rajasthan, India. The animals were irradiated at a dose rate of 0.95 gray per minute during the first year and 1.85 gray per minute for the subsequent year. So mercury salt. Actually, we use the mercury salt, not the mercury. We use the mercury chloride of analytical grade was used for the present study. It was purchased Brent Baxi Laboratory Limited from India. It was administered orally in drinking water at the dose of 0.5 parts per million. So plan was the experimentation was, the animals were divided into the 7 groups. So group 1 was the sham irradiated animals, normal conditions. Group 2, mercury chloride treated animals. Group 3, only irradiated animals, which has the 2 subgroups. Group 3rd A, 2-point grade, and the 3rd B group was the 4-point grade. 4th group was the combination of radiation and mercury chloride. It has also 2 subgroups. It had also 2 subgroups. 4th A, 2-point grade plus mercury chloride, and 4th B, which is 4 grade plus mercury chloride. Group 5th, mercury chloride plus moringa. 6th group, radiation plus moringa, and group 7, radiation plus mercury chloride and moringa. So, autopsy of animal. 5 animals from each group were autopsied after 1 to 4, 7, and 14 and 20 days of the treatment. The animals were sacrificed by the cervical dislocation. So, parameters, the following parameters were taken into the consideration. First is the histological studies, and second is the biochemical studies. So, histological studies has a qualitative and quantitative. The jejunum was fixed in the Goins fluid, and the process for the histological observation by routine procedure, and the parameters studied were as follows. Qualitative changes: histological changes in various layers of the intestine, including the crypts and the villi. So, quantitative changes: length of villi, number of goblet cells in crypts and villi, number of mitosis in crypts, number of abnormal mitosis in the crypts, number of dead cells in the crypts, and number of total cells in the crypts. Biochemical studies were the following parameters like total proteins, cholesterol, acid phosphatase activity, alkaline phosphatase activity, DNA and RNA. Results: biochemical, the value of the total protein, cholesterol, DNA and RNA decreased up to the 14th day in non-drug treated groups taken 3 and 4, and day 7 in the up to the day 7 in the moringa treated groups 5th, 6th and 7th. Thereafter the value increased on the day 28 in non-drug treated groups, and the day after the day 14 in the moringa treated groups. The value of acid and alkaline phosphatase activity were increased up to the 14 days in the non-drug treated groups, and day 7 in the moringa treated groups, then declined in the all groups. The severe changes were observed after the combined exposure, showing the synergistic effect and early recovery, and less severe changes.
Sorry.
Yes.
Please come to the conclusion.
Okay. histology. So these were the conclusions. After the combined exposure of gamma rays and mercury chloride, the histological changes were similar but showed the higher magnitude than the individual exposure of the gamma rays and mercury chloride. The jejunum of the moringa treated animals exhibit less severe damage as compared to the non-drug treated animals at all the corresponding intervals. In early and fast recovery was also noticed in the moringa pre-treated animals. So these are the photographs of the normal structure. These are the normal jejunum. And then photograph of the jejunum of the Swiss albino mice after 14 days of mercury chloride treatment displaying the hydroponic degeneration in the villi. This is the photograph of the jejunum of Swiss albino mice after 2 days of the gamma rays exposure, illustrating that was a 2-point ray, illustrating abnormal mitosis and anaphase bris, cytoplasmic degranulation and evacuation are also visible. These are the 7 days. After the 7 days of gamma radiation with the 4. And after 4 days of the combined treatment of gamma rays and mercury chloride displaying the severe damage, cytoplasmic degranulation, vacuolation, and necrotic cells are also seen. Pycnotic nuclei and abnormal mitosis are evident. After 14 days of gamma ray exposure with the prior administration of the represents the mild cytoplasmic degranulation and increased number of mitotic figures. After 28 days of the exposure of the gamma rays, showing the increased number of the goblet cells in the villi. So conclusion, from the present findings of the following conclusions could be drawn. The jejunum of the Swiss albino mice suffered with the radiation and mercury chloride at the histological, mercury induced changes at the histological and biochemical levels, alteration in the histological structures followed by the biochemical changes. The mitotic figure showed the parallel behavior with the total cell population and the inverse relationship with the number of dead cells. The abnormalities in mitosis related to the cell death. The combined treatment of the radiation and mercury chloride showed the synergistic changes. And the jejunum of the moringa treated animals showed less severe radio lesions and an early and fast recovery in the comparison to the non-drug treated animals. Thus, it seems that moringa has protected the jejunum at both the dose level. The moringa might have protected the animals from the radiation by more than one mechanisms due to the multiplicity of its properties. So moringa protects the animal by increasing level of glutathione. It also neutralizes the peroxide from the water molecules after the irradiation. Thus, the moringa is a good herbal radioprotector and can be given to cancer patients during the radiotherapy to minimize the side effects of the exposure. Thank you.
Is there any question?
Hello. Pooja, may I know how you measured the quantity of peroxides after giving moringa?
Can you repeat the question, ma'am?
How you estimated the quantity of peroxide radical?
I didn't get the question. Quantity of which?
आपने peroxide radicals को किस तरह से measure किया? Enzyme activity देखा था या peroxide radicals देखा था?
Alkaline phosphatase activity and the acid phosphatase activity.
Alkaline phosphatase and acid phosphatase.
Measurement of peroxide.
What is their role in?
No, ma'am. ये वाला मैम peroxide का तो नहीं दिया मैंने सिर्फ हाँ.
आपने लास्ट में बोला ना कि peroxide radicals को भी कम करता है मोरिंगा.
Yes, ma'am.
न्यूट्रलाइज करता है उसको भी. तो peroxide radicals को कैसे मेजर किया था आपने पहले और बाद में?
That was ये है कि जब हमने उसको रेडिएशन दिया था, तो जब हम रेडिएशन देते हैं तो peroxides बनते हैं. So for that मैंने ये लाइन ऐड करी है उसमें कि peroxides जो हैं वो reduce हो जाता है.
ये मेजर किया होगा ना?
हाँ, मैंने कोई मेजरमेंट नहीं किया, कोई एस्टिमेट.
May I ask a question or a comment rather?
Yes, sir.
Yeah. There was very detailed works are also being done by a group from Calcutta University, Professor Sanjit De, on moringa and quercetin. And they have also shown the radioprotective properties, which you may get it in, I think, IGRB. Okay, my one of the observation is that you have also included mercury chloride in your studies.
Yes, sir.
So it's itself a very, 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 very toxic.
Yes, it's very toxic.
Compound. Why did you use it, and what was the result of your toxicity studies on the animals? I mean, how did you decide what kind of concentration you are going to use?
Actually, they have set an experiment for animals कि हमें कितना how much मतलब हमने उसका experiment dose लिया कि कितना हमें mercury chloride किस level पर जाकर toxicity जो है वो जो animals है वो sustain कर सकते हैं उस toxicity को.
Yeah, Pooja, Dr. Srikanth is very senior scientist from IUC New Delhi. What he means to us, I want to mention here. He's asking you have decided the dose of mercury chloride.
Yes, sir.
Suppose you go 1 p.m. or 2 p.m. So it is a very toxic element.
Yes, it is.
How did you select the dose? One question about that.
Yes.
And the comment mentioned by Professor Benki that a team from Calcutta, I know that they are still working on the moringa.
Yes.
Can you go through their research papers, articles, how they have decided the doses? And keep it in the mind of Srikanth from IUC, what he has mentioned.
Yes,
you should give the proper answer. You are presenting your paper in interesting conference. How to decide the dose of mercury, how to decide the dose of radiation. Dr. Srikanth, I think you're going to ask this question. No? How did you decide?
Yeah. I mean, I just wanted to know because it's a very important study. And also another point is that she mentioned that using moringa during the radiation therapy would also reduce the adverse effects. But then the radiation therapy of the cancer cells, they will also get the protection from the moringa. Okay, so is there any study planned to see the differential effect?
Yeah, I think what they are doing, Dr. Srikanth, they have divided the animals into different groups, like only moringa, like only mercury, like only radiation, and they are the control groups, and along with the moringa, they are the experimental groups. Compared study she has done. They have started administering the moringa oleifera extract 7 days prior to the irradiation, and the extract was continued till the last autopsy interval. This is what I don't understand.
Okay. Okay. Thank you. I think a very good collaborative work can be done along with Sanjit.
Yes.
He does lots of these things.
I know Dr. Sanjay. I know we have all the papers, and I always advise them to include his papers in the offices also.
Yeah, sure, sir.
Yeah.
Please take into consideration Dr. Srikanth, a very senior scientist from the field area. Okay. Thank you very much. Thank you very much.
Thank you.
Okay, thank you very much.
Hello. Hello. Can I add a point if I have the permission?
Yes, Dr. Kalia, please go ahead.
See, this study includes mercury as far as potentiation of radio sensitization effects are concerned, and then the protection by whatever compound you use. But then the conclusion says that it is a radio protector as far as radiation therapy is concerned. Then what is the rationale of using mercury in these studies when in conclusions or implications radiation plus mercury plus moringa is not discussed? It's a curiosity, that's why I asked. maybe somebody else will have some explanation.
Dr. Kalia, you have mentioned correctly, it should have been included in the conclusion. Pooja, it should have been included in the conclusion. What Dr. Kalia is, please be seated. What Dr. Kalia has mentioned, that in your conclusion, as you have mentioned, the only mercury, only radiation, combined treatment, and along with the moringa. But your conclusion is insufficient. It should have been taken into consideration. It should have been taken into consideration. So you take in the future when you submit your thesis, you have to look after all the things. Thanks, Dr. Kalia.
Thank you, Pooja. Our next speaker is Anushka Khanna from INMAS, New Delhi.
Yes, ma'am. Ma'am, am I audible?
Yeah, you are audible.
Anushka, please stay in your limits, time limits.
Okay, ma'am. Ma'am, is my screen visible?
Yes, your screen is visible. Continue.
Good afternoon, everyone. I am Anushka Khanna, working under the supervision of Dr. I. Prem Kumar at INMAS DRTU, and I'm here to present my work on the topic short-term exposure of bone marrow cells to a potent MPTP inhibitor enhances hematopoietic stem and progenitor cell migration, homing, and repopulation potential. Basically, my work focuses on the management of hematopoietic form of acute radiation syndrome. So it is a possibility to manage it, provided it is managed at an early time point. Upon radiation exposure, the loss of HSCs results in the inability of the system to generate the differentiated lineages, which actually compromises the immune status, which paves the way for opportunistic infections, which is the major cause of death in case of bone marrow syndrome. So as of now, hematopoietic stem cell transplantation is the most well-established and widely accepted modality for its management. It is of use when it is sure that the bone marrow of the victim is extensively compromised. There are certain limitations to its use. For example, identifying the right HLA matched donor in a given time frame, because there is no sufficient time window after radiation exposure. So recently, for umbilical cord blood have been found to be useful in such scenarios, since it reduces the chance of rejection. Although there are certain drawbacks to its use, like the number of stem cells per unit cord blood is very low. So various strategies have been employed to address this issue, like expansion of hematopoietic stem cells ex vivo, and or either by improving the homing efficiency of the transplanted cells. Because to achieve successful transplantation, it is important that sufficient number of stem cells are infused. So these are various strategies that can be employed to improve the homing and subsequent engraftment. So in my research, the following approach I have used. Basically, stem cells reside in hypoxic niches inside the bone marrow. So the transition from the hypoxia to the normal environment where all the collection and modulation of stem cells takes place, it leads to ROS production, oxidative stress, and opening of the mitochondrial permeability transition pore, which leads to rapid and reversible changes in the metabolism of HSCs. Which is known as extraphysiological oxygen stress and shock, and this has a lot of negative consequences on the survival and long-term repopulating potential of the stem cells. So in the approach that I've used is I've screened various drug libraries to identify a potent MPTP inhibitor along with their capacity to enhance the CXCR4 expression, which can together improve the homing and subsequently the transplantation efficiency. So 3 screening strategies I have employed, and following the primary, secondary and tertiary screening, I have shortlisted a few molecules which proved to be potent MPTP inhibitors, and amongst them, some of them had negative consequences on the stem cell surface marker expression. So sodium hydrogen sulfide, which is a H2s releasing molecule, along with its many properties like antioxidant properties and radiomodulating properties, it is also readily available and cost effective. So I have taken this forward for all my experimental work. So the following preliminary studies in my lab, the following strategy has been laid down, in which the bone marrow cells were isolated from mice, and then they were exposed to this sodium hydrogen sulfide for a particular time period at 37 degrees Celsius, and then subsequently, all the homing studies and repopulation and in vitro studies were studied in this LSK population and the long-term HSC and STHSC. This is the gating strategies that I have employed for rest of the experiments. CXCR4 expression is very important, as the CXCR4 is expressed on the hematopoietic stem cells, and the CXCR4 and SDF1 protein, which is expressed in the stromal cells, is the fundamental mechanism which guides the migration of stem cells from the circulation to the bone marrow niche, which is important for efficient homing. So this NAHS exposure resulted in significant increase in the both the gene and protein expression of CXCR4 in different subsets of hematopoietic stem cell population, which I have measured through flow cytometer. Toxicity studies were done, and this molecule did not induce significant toxicity at the concentration and the time point that I have chosen for the studies. Next, in vitro transfer migration was performed in order to assess the effect of NAHS treatment on the migration of stem cells, and the cells were incubated in the presence and absence of SDF1, and the cells were enumerated through flow cytometry. NAHS exposure resulted in a significant migration of the stem cells towards the SDF1 alpha, which was significantly inhibited by the CXCR4 antagonist AMD 3100, which proves that this enhanced migration was due to the enhanced CXCR4 expression. Apart from CXCR4 expression, the expression of adhesion molecule integrin 6 alpha was also monitored and measured through flow cytometry in different subset population of hematopoietic stem cells, and NAHS exposure also enhanced the surface expression of this adhesion molecule. Apart from this, cell proliferation was also monitored, and using PI staining and Ki67 cell proliferation marker. So the results indicate that the NAHS exposure resulted in the enhanced cycling, in the enhanced number of cycling LSK population after the treatment as compared to the untreated control. The cell proliferation marker expression, the results also corroborated with the cell cycle status, and NAHS exposure resulted in enhanced expression of Ki67 marker as well at 24 hours time point. Since EFOS or MPTP opening results in the loss of colony forming potential. So since this is an MPTP inhibitor, the colony formation was assessed through microscopy after 14 days, and this was the methodology that was followed. And the colonies were counted, and it showed that the NAHS had a positive effect on the colony forming potential, and it enhanced the colony forming potential of the stem cells. Apart from this, nitric oxide signaling plays an important role in CXCR4 expression. So this NAHS exposure also had a positive effect on the nitric oxide production. Then next, the intracellular calcium levels were monitored. This was done because CXCR4 calcium signaling is very important for the upregulation of CXCR4 expression. Since NHS exposure resulted in both the enhanced proliferation and CXCR4 expression, the intracellular calcium levels were monitored in the presence of NAHS, and it significantly enhanced the intracellular calcium levels, which was measured through flow cytometer at various time points. And then along with that, CXCR4 protein and gene expression was monitored in the presence of calcium chelators, and thus in the absence of calcium, the CXCR4 expression reduced. Then next, the role of calcium mitochondrial pathway was assessed through monitoring the mitochondrial activity after NAHS exposure, and it was observed that sodium hydrogen sulfide significantly enhanced the mitochondria and modulated the mitochondrial activity, thus indicating that there might be a role of calcium mitochondrial pathway which can be attributed to the cell proliferation. Finally, in vivo studies were performed in which the stem cells were isolated from the mice, and then they were treated as I have mentioned above, and then they were transplanted into lethally irradiated mice, and after 24 hours, the CXCR4 expression and the homing was monitored. The results corroborated with the in vitro studies that were performed earlier, and NHS exposure resulted in enhanced CXCR4 expression, and also the number of stem cells, stem cells that were treated with NAHS, more number of stem cells reached the bone marrow of the recipient cells, which can be seen through this graph, and it was measured through flow cytometry. Then the effect of sodium hydrogen sulfide on the repopulation potential was monitored. Similarly, the cells were treated and they were transplanted into lethally irradiated mice. And just like the in vitro studies, after 24 hours, the NHS treated cells displayed more number of cells in the cycling state as compared to the untreated ones. And after 30 days, the hematology profiling was done, which also indicated that it significantly enhanced the repopulation. The NHS exposure significantly enhanced the repopulation potential of the stem cells. So the in vitro and the in vivo studies demonstrated that NHS exposure proved to be a potent MPTP inhibitor. Along with that, it also resulted in enhanced CXCR4 expression and homing and repopulation potential of the stem cells. This can be attributed to the increased calcium influx, thus activating the mitochondrial pathway, resulting in all the downstream changes. Overall, this study proposes that short-term ex vivo exposure of bone marrow cells to sodium hydrogen sulfide might prove to be an efficient as well as cost-effective strategy to achieve successful transplantation. These are the publication and references. Thank you.
Thank you, Anushka, for your nice presentation. Any question? Okay. Our last speaker is Dr. Tina Haritwal from INMAS. Dr. Tina, are you there?
Yes, ma'am. Just a second.
Dr. Tina?
Yes, ma'am.
You have just 5 to 6 minutes.
Okay, ma'am.
Dr. Anushka, please stop sharing your slide now.
Yes, ma'am, I'm trying.
Ma'am, just a minute.
Yes.
Okay.
Tina, is there any problem?
Just a ma'am, just a minute, ma'am. Am I audible, ma'am?
Yes, you are audible. Your screen is not visible. Okay, continue. Tina, please continue. Tina, please unmute yourself.
Am I audible, ma'am?
Yes, you are audible. Please continue.
Okay. Ma'am, I'm Dr. Tina Haritwal from INMAS DRDO, and my today's presentation entitled as Diallyl Sulfide Mitigates Radiation.
Dr. Tina, probably you are using two devices and both the devices have their mic on. So it is echoing.
Now is it better?
Please start.
Tina, are you there? Dr. Tina Haritwal.
Ma'am, yes, ma'am. Yes, ma'am. Am I audible?
Yes, now you are audible.
It is okay now.
Okay, just a moment, ma'am. I have to share my screen.
You have simultaneously joined Zoom with two devices. That's why echo is there at your end. So please join through one device only.
Ma'am, is my screen visible?
Yes, visible.
Okay, thank you so much for the introduction. And I'm Dr. Tina Haritwal from INMAS DRDO, and my today's presentation entitled as Diallyl Sulfide Mitigates Radiation-Induced Damages as Evident from NMR Metabolomics and F1 generation. My seniors and my colleagues have already told a lot about radiation countermeasures, so I will not elaborate this. My main focus is on radiomitigative agents, which are administered shortly after radiation exposure or within 24 hours of radiation exposure. So basically, I'm using diallyl sulfide as a radiomitigative agent, which is a very well-known organosulfur compound, which is extracted from garlic, and it is a natural compound which is being used as a histone deacetylase inhibitor, and it has anti-cancerous and anti-apoptotic activity. As I have already told that there are various histone deacetylase inhibitors which are being used as anti-cancerous agent, and diallyl sulfide is a dietary supplement which is very well known and reported histone deacetylase inhibitor. We are using it as radiomitigator agent. My main focus is on reproductive system. So radiation doses even low as 0.15 gray result in oligospermia, means reduced number of sperm cells, and doses more than 0.5 gray result in azoospermia, means diminishes fully sperm cells, and it causes temporary or permanent sterility. So if the mutation occur in germ cells, transmit from parents to their progenies, then there is chances of genetic diseases. So basically, due to radiation exposure, there will be changes and damages in DNA and proteins, and by that, cell death will occur more. So there will be a reduced number of germ cells, and birth defects and anomalies in progenies will occur if it transmit from parents to their progenies. So to solve this problem, we have a simple hypothesis. Here is the silent gene shown in the picture. Histone acetyltransferase enzyme histone acetyltransferase enzyme will transfer its acetyl group to the histone tails, and the gene will be in relaxed form, and gene expression will be enhanced. After a certain level of expression, histone deacetylase enzyme will come and convert it into the silent stage again. If we administer histone deacetylase inhibitor, then due to hyperacetylation, gene expression will occur more. Just after radiation exposure, if we administer histone deacetylase inhibitor enzyme, then due to hyperacetylation, DNA genes will be in relaxed form, and there will be more site to the to repair protein accommodation, and due to that, there will be enhancement in the repair of DNA damage. So for that hypothesis, we have taken C57BL6 mice. 2 gray of gamma radiation was given, and diallyl sulfide was used as drug. It was administered through IP, intraperitoneal, and 2 hour post irradiation. And all of the experiments were conducted 24 hours, 3 weeks, 5 weeks post irradiation. First, we need to study that DAS is working as SDAC inhibitor in testis or not. For that, we have done 2 assays, SDAC inhibitor, SDAC activity assay, and DNS1 accessibility assay. In SDAC activity assay, we have calculated deacetylated lysine content, which was significantly increased by 2 gray of gamma radiation exposure, and it restored back to the normal level by diallyl sulfide administration. And a similar type of similar type of results we have found in DNS1 accessibility assay. It is performed to gain insight of open chromatin after various treatments, and diallyl sulfide has shown a better smear, which shows that there is a better repair in the damaged sites. So these both studies have shown that DAS has SDAC inhibitor activity in mouse testis. So further, we have to check the mitigating potential of diallyl sulfide. We have done some preliminary studies as testis body weight index, as sperm head abnormality assay, motility assay. It's an image of which shows various sperm head abnormality, and there was a very reduced the lowest body weight. Testis body weight was recorded at 3 weeks post irradiation, which may be a result of loss of various spermatogenic cells, and it was recovered by diallyl sulfide administration. Sperm head abnormality assay is a good indicator of fertility, and it is being used to check the genotoxicity of various mutagens. So we have found that the highest number of abnormal sperms were recorded at 5 weeks post irradiation, and similar results were found in motility assay, which the restoration of DAS, restoration of motility by DAS shows the intactness of DNA and intactness of sperm membrane. So these all studies shows DAS has shown a better result in preliminary studies. So further, we have done viability assay by two methods. First is tap and blue, and second is using PI uptake by flow cytometry. And we have found similar type of results in both of the studies. Both studies are in corroboration of each other, where we found that highest number of non-viable sperms were recorded at 5 weeks post irradiation, which were recovered by diallyl sulfide administration. And here in this image, you can see that the last I cannot use my marker actually, but in the quadrant 4 is showing the highest. Quadrant 4 is showing non-viable cells in the dot plot, and where we can see that quadrant 4 of radiation group has shown the highest number of non-viable cells, which was recovered to the normal level by diallyl sulfide. To further confirm these studies, the gold standard of all the preliminary studies, histological parameter analysis. Here you can see that these images are of 3 weeks post irradiation histological study of testis, and we can see that the beautifully arranged seminiferous tubules with intact basal membrane, with beautifully arranged spermatogonia cells and other spermatogenic cells, and lumen is filled with spermatid cells. And 2 gram gamma radiation exposure has shrunken the seminiferous tubules and created a cavity in the seminiferous tubules and lost all the spermatogenic and spermatogonia cells, which were recovered by diallyl sulfide administration. Some of the lumens are filled with spermatid cells, and some of the seminiferous tubules are other spermatogenic and spermatogonia cells. We have done. We have basically recorded. We have recorded these all 3 types of cells by microscopy also, and we have found that all 3 types of cells were lost at 3 weeks post irradiation. Spermatid cells remained. Spermatid cells remained lost after 5 weeks post irradiation. Diallyl sulfide administration has significantly restored all of the number of cells. To further confirm this study, we have done cells 3 weeks post irradiation. As I can show you here, you can see that in the radiation alone group, spermatogonia cells, histogram of spermatogonia cells has been lost fully, and which was recovered to a certain level by diallyl sulfide administration. And it is seen in this study is in corroboration with the previous histological study. These both study has shown similar type of results, where we can see that all of the cells were reduced after 3 weeks post irradiation, which was recovered back to the normal level by diallyl sulfide administration.
Tina, kindly conclude.
Okay, sir.
We have to start next session also.
Yes, sir.
Session still. When you see the scientists, they are waiting.
Okay, sir. So next is metabolomic alteration. Metabolomic study was also conducted by using NMR, and where we can see that there were certain metabolites were altered due to radiation and due to radiation exposure and diallyl sulfide administration. Adenosine, which is responsible for energy metabolites, and high level of energy is required for spermatogenesis, and altered level of adenosine may be responsible for the viability and loss of viability and sperm abnormality. Myoanisitol and taurine, these two are responsible for, these two are osmolytes are responsible for membrane integrity. So they may be the reason behind the sperm head abnormalities. And choline and phosphatidylcholine, these two metabolite alteration may be responsible for motility, because these are in abundant number in the sperm head in the membrane, and betaine is responsible for hyperacetylation. So DAS has significantly changed these all to the normal control values. Further, we have done studies in F1 generation also. 2-graph gamma radiation was given to the mice, and 2 hours post radiation diallyl sulfide was administered, and 24 hours 5 days post radiation. These treated animals were breeded with untreated female.
Come to the conclusion.
Okay. And hematological studies were also. So in F1 generation also we have seen this results. So my conclusion is due to exposure of radiation, altered level of metabolites will occur, and higher number of abnormalities in sperms, and viability and spermatogenesis both will be lowered, and by that reduced fertility and genomic instability in next generation will occur, and it ultimately causes to radiation induced hematopoietic depression in F1 generation, which is basically shown by the hematological parameter studies in F1 generation. And diallyl sulfide has reduced the radiation induced reduction in spermatogenesis and altered level of metabolites, which ultimately mitigates radiation induced hematopoietic depression in F1 generation. I would like to thank the director INMAS and mainly to the organizers of this conference, and thank you so much.
Professor Venki, for your kind words of blessings. You are invited. Chairperson Dr. Venki.
Good evening, sir.
Very good evening. Very good evening.
Yes, sir. So thank you for the presentation. I think the session, there are 6 presenters, and all the presentations are extremely good. Yes, Professor Kalia was mentioned, the online as well as in physical presentation, we are seeing its own advantages as well as disadvantages. Despite the limitations, all the presenters made their best effort to give a good presentation within the stipulated time frame as well as with the clarity. Okay, thank you so much. Thank you.
Thank you, Venki, for your blessings. May I request Madam Veena B. Kosara, can you say a few words of blessings to the young researchers?
Thank you, sir. Thanks for giving me this opportunity. I really like the presentations given by the students, and I wish good luck to them. Results you will be announcing tomorrow, so I need not say anything more.
Okay.
Thank you, sir. My good wishes are always with you.
Thank you, ma'am. Madam Madhuvala is there, the President Society.
I have been there throughout. Don't worry.
Yeah. Welcome, ma'am. Please say a few words of blessings to the researchers.
This young scientist session is always very close to my heart, and like previous conferences, this has been a scintillating session. All the presentations have been made with great fervor. Students own their work. That is very important. And of course, because of paucity of time, this number is less, but competition is not less. So I wish them all good, and best of best wishes. Keep doing good work. Tomorrow you will get the results, right? I'm told that, but I will leave it to the audience.
Your blessings
Announce the
matter
results.
for all the young scientists, including me also. May I request Professor Kalia, at this age, I am very happy to see since morning he has been with us. In all the censuses, he has interacted very nicely. Kalia sir, you also say a few words of blessings to the young researchers. Professor Kalia.
Sir, please switch on your mic.
Sir, kindly unmute yourself. Dr. Kalia, you are not audible. Kindly unmute yourself. Professor Kalia. Yeah.
Am I audible now?
Yeah.
Okay. I'm not very tech savvy. I just forget these things. See, as Dr. Madhubala already said, all the presentations were very good. One thing is excellent work done with dedication, good experimental design, good data, and they are learning the present art of presentation in a limited time, which is very important. When I pointed out also, this is essentially the work possibly from your lab, cadmium and mercury, along with radiation. I pointed out in the Viva also when I attended in person for one of your students.
I do remember.
So we will discuss that later. But the point is, when that such a parameter is included, what is the rationale of including that parameter? And what are the implications of that? Where is the applicability of that? When you say radiotherapy, radioprotection, then where does mercury or cadmium fit in the picture? That was my point. But nonetheless, I said in the Viva also, that is not the student's limitation. Student is a student, a beginner will do what the guides tell.
Yeah.
So as far as the students are concerned, as Dr. Madhubala said, they have made excellent presentations, and whosoever are the judges will have tough time in deciding who should be awarded. The other limitation what I realized was, I asked this question, that one of the presentations, the slides were not coming out, I mean, like they should be coming on the screen. Then I was told, sir, she is presenting through the mobile phone.
Okay.
Similarly, I ran into the problem that my laptop developed some problem. Then I had to on the whole passcode and everything I had to undergo and then come back. So these are the limitations.
Thank you. Thank you, sir. Thank you very much. Thank you, Professor Madam Madhubala. Thank you, Kalia sir. Thank you, Dr. Venki, Dr. Veena B. Kushaba. Thank you, Dr. Archana, very active faculty member of the department. Ma'am Chetana, we also welcome you. I would request Dr. Kalia, you met in Bikaner, Dr. Shishir Sharma. At that time, he was here in our college. He is in the Department of Geology. Now he has become principal of Government MS College Bikaner. He has been with us since long. So I would request Honorable Principal Maharani Sudarshan College, Dr. Shishir Sharma, along with his life partner, Dr. Rajni Sharma, the Professor of Economics in MS College. You both are requested to come to the dais. I would request Dr. Deepti, Dr. Aruna to felicitate our Dr. Chetana and Dr. Archana by giving certificate and memento. The keynote speakers and the chairpersons joining online, we will send the certificate and memento by post. Welcome, Dr. S. Sharma, Dr. Sharma, Dr. Anila, you also come.
Dr. Purohit, along with that, you please send the meals also. You must be having delicious
Sure, Dr. Madhubala, sure. Meal will be awaited. I will invite you all. And then when the COVID situation gets normal, I will invite you all.
That is what I was going to say, Dr. Purohit.
Ma'am, due to technology, even we didn't miss you. And we are so happy you have been with us in all the sessions this morning. I know your busyness, I know your health problem also, but you all work with me. I am so happy, madam. You always promote Dr. Kalia and Venki and Madhubala ji. They always promote the radiation biology from Bikaner. You are so popular in Bikaner media.
Yes.
Bikaner media.
I have some ideas which I thought I will come up with if I were to be physically present.
Sure, sir.
For Bikaner, I had some ideas in mind, but anyway, maybe in good times we will discuss.
One more thing, Dr. Kalia, we are missing your that brown coat. We are missing your. Always you wear that red coat.
Oh, my maroon coat.
Yeah.
My maroon coat.
Maroon coat.
Yeah. I'll take it out today.
My student was asking.
Okay.
Okay. Our honorable principal, MS College, Dr. Shishir Sharma. Dr. Archana, actually look at him, just I just congratulations. Sir, yeah, Dr. Chetna, the chairperson and the director and the owner, Gurukul College, Ponsongarh, with the district of Ganganagar. She has come since yesterday morning. She has attended all the sessions. She has chaired the sessions. She is one of the invited speakers also. I would request Chetna to say a few words. Dr. Sisi Sharma will also speak few words of blessings to all the delegates.
First of all, thanks for inviting me. It's a big news. Congratulations to Dr. Rohit and his team for great grand success of this ICRD. Thank you all of you. Jai Bharat, Jai Hind.
Sisi Sharma जी हमारे बीच हैं। बड़ा केवल कहना बड़ा अच्छा लगता है कि एमएस कॉलेज आए, लेकिन हम इनको यही का मानते हैं डूंगर कॉलेज का ही। आज भी सर वर्षों साथ जुड़े हुए हैं, 3 दशक हम साथ रहे हैं बीकानेर। मैं सर से आग्रह करूंगा कि विद्यार्थियों को, फैकल्टी मेंबर्स को अपना आशीर्वचन देंगे। डॉक्टर सीसी शर्मा सर।
थैंक यू राजेश जी। सबसे पहले तो आपको इस शानदार आयोजन के लिए बहुत बहुत बधाई। और ऐसा लग रहा था कि ये आयोजन 20 में होना था, इसमें तो 22 हो गई, लेकिन जब इन्होंने किया है तो पुराने सारे कीर्तिमान वापस ध्वस्त कर दिए। इस कोविड कंडीशन में, इस हाइब्रिड मोड में अंतरराष्ट्रीय सेमिनार कराना और उसको सफलतापूर्वक कराना, हाईटेक कंडीशन बीकानेर में वो उपलब्ध होना, ये मेरे को लगता है शायद रायन जी की काबिलियत है। और जैसा कि बराबर कहते आ रहे हैं कि अब मैं चाहता हूं कि हमारी और फैकल्टी मेंबर्स भी थोड़े से उत्साहित हों और इस तरह के अंतरराष्ट्रीय सेमिनार वेबिनार कराएं। मेरे को लगता है कि अब इन्होंने बैटन शायद नेक्स्ट को दे दिया होगा या इस सेमिनार के बाद दे देंगे। इनके सुखद भविष्य की मैं कामना करता हूँ। ये भले ही सेमिनार यहाँ ना करें, लेकिन ये समाज में और भी कई तरह के सेमिनार्स कराएंगे, ये मेरे को मालूम है। तो एक बार पुनः इस सफल आयोजन के लिए इनको बधाइयाँ। हालांकि आज दूसरा ही दिन है, कल तीसरा दिन भी है, वापस इनको बधाइयाँ मिलेगी। और मैं भी हूँगा, ऐसा नहीं कि मैं आज इनको कह के ही जा रहा हूँ, कल मौका मिलेगा तो वापस यही शुभकामनाएं वापस कहूँगा। लेकिन एक चीज मैं अभी कहना चाहूंगा, मैं एमएस कॉलेज चला गया, ठीक है, सरकारी प्रक्रिया है, जा सकते हैं, लेकिन मेरा दिल, मेरा दिमाग, मैं यही कहूँ 35 साल मैंने इस कॉलेज के अंदर जॉब किया है। इस कॉलेज की एक एक होना क्या है ये मेरे को पता नहीं। मैं यही सोचता हूँ कि मेरे 2 कॉलेज हैं, एमएस कॉलेज और डूंगर कॉलेज। मैं बहुत भाग्यशाली हूँ, मैं 2 कॉलेज संभाल रहा हूँ। और पुरोहित जी को बहुत बहुत धन्यवाद कि आपने मुझे सम्मान दिया। थैंक यू।
We all have heard honorable principal of this college, Dr. Sisi Sharma, the great scientist, great geologist. Ladies and gentlemen, one session on the radiation cancer. We are going to start I know the already more than 5 p.m. I would request the chairperson of the session that is cancer, my fellow colleague, senior faculty member, and the co-convener of the this organizing committee, Dr. Dipti Srivastav. I would like to introduce Dr. Dipti Srivastav. Madam Srivastav, you come on the dais. Dr. Srivastav, he has produced 4 PhDs under her supervision. She has visited Mauritius and delivered her talk, and the Best Presenters Award. She is the recipient of Best Presenters Award in the Mauritius, and she's a great entomologist. She's working on different kinds of insects, pesticides, and she's one of the I can say vertebra of this ICRB 2022. Without caring over health, she has been with us since long, since right from the morning. Suppose I call at 9:30, she reaches here at 9:15. We all should learn lesson. She is the eldest in department zoology among all. Ma'am, I pay regards on behalf of our department. Thank you so much. Ladies and gentlemen, one of my young colleague, very active, very enthusiastic colleague, Professor Jaishankar Acharya, Professor of Chemistry. I would request Dr. Jaishankar to come on the dais. He also will chair the session. Dr. Jaishankar is also the recipient of many awards from the Rajasthan Patrika and other from media. She has also produced many papers, attended various national international conference, chaired the session. She is man of chemistry. He is the man of chemistry. I would request one more chairperson along with the rapporteur, the one of the vertebra of this ICRB, the member of Executive Council in the Society for Radiation Biology, Dr. Aruna Chakrathi. Dr. Aruna, come with the dais. Dr. Aruna Chakrathi, she has produced around 10 PhD under supervision, around 15 MPhil. She has also attended many national international conferences, published many research papers, and the third consecutive time she is the organizing secretary of the international conference. Ladies and gentlemen, Madam Vibha Tandon is there. Professor Vibha Tandon.
Yes, I am there.
Madam, I am very sorry we are too late. Vibha Tandon, Madam, I am so impressed with you. We have been together in many conferences. Last time we were at Mangalore, and she always motivates young scholars, young scientists. Ma'am, we are proud of you. Since long you have been waiting for us. And now over to Dipti Srivastav to invite Vibha Tandon. Then senior scientists from INMAS, then the Gorakhpur. They will be talk. Please, I request audience to please be with us. You will enjoy very interesting talks will be going on. Dr. Dipti Srivastav, please go ahead.
Thanks, sir. And thanks, organizing committee, for giving me this opportunity. Now, first of all, I welcome you all in this sixth technical session. Now, I invite Dr. Vibha Tandon from New Delhi to deliver her talk.
Okay, thank you very much, and thanks a lot for the short introduction. I would like to thank Dr. Madhubala, who always have a smiling face, long time no see, but obviously we remember her, and I crossed her 2 years or 1 year before on airport somewhere, and we had waved each other. So, but I would also say that I would like to really congratulate Dr. Purohit and his team. And they have done a wonderful job. And I was also listening. It's not the young scientist lectures. And I was thinking that we should not be involved in giving the lectures. Actually, all these students should be given the opportunity, because those were wonderful lectures. I would really appreciate the courage shown by Dr. Purohit and guidance shown by Dr. Madhubala. And I was also supposed to do something for ICRB, but ISRB, but I couldn't do it. And I would like to apologize on the
No apologies, ma'am. No apologies. You still have the time. Please go ahead.
But still, thank you very much. And I won't take a time, and I will try to go ahead very fast.
No, we will not accept your apologies. You have still to do it.
Yes,
Okay.
I will do it. I will do it, and I will certainly do it. And this time I will not say, not show a sorry face to you. So thank you very much. And so the title of my talk is the Prochlorperazine enhances radiosensitivity of non-small cell lung carcinoma by stabilizing GDP-bound mutant KRAS conformation. I could see that there is a mistake in the title. I would like to, can you see my slides, you people? Everybody is able to see my slides?
Yes, yes, we have seen.
Okay. So I will be going quite short in my lecture because I know that the whole program is quite late. So the whole essence of my lecture is this, that we have tried to develop a radiosensitizer which happened to be the KRAS inhibitor also. So we have taken an approach here which was somewhere started some 4 or 5 years before, a committee of the somewhere in the ISRB, only the say NIH people came, and they wanted that we should work on the KRAS inhibitors as the radiosensitizers. And that is what was this theoretical work is started, and a few of the labs were given the work to look into the some of the drugs which can work as a KRAS inhibitors also. And because the KRAS resistant cell lines, resistant cells become the radio resistant also. So the whole say motto of this work was there, that if we can make a molecule which can inhibit the KRAS also, and so it will be able to work as a radiosensitizer also. So on the basis of this, we could this whole theory started from there. And while we were doing the radiotherapy for the today, work, when we started the radiosensitization and radioprotection, which we have been doing in 2003 or 4, we have started, there was a whole plethora of work was done by 4 different labs in the world, and they have given us a series of the molecules which can work as a KRAS, which act on the KRAS different 4 drug binding sites of the KRAS. And these drug binding sites of the KRAS were actually shown that they have shown it that they are having a very good inhibitory activity. And one of the drug binding site which they have chosen was the same site where the GDP used to bind. So this, as all of you know, that the KRAS is a GTPase protein, and GTP has a very high affinity to bind to the KRAS. And if you want to replace GTP by some other drug, it is just a I would say it's not a good research, but yes, we have done it, and we have tried to do it, and I wanted to give it a try, and we spent some last 5 to 6 years with us on this different molecules. And what the data I'm presenting here is on the say prochlorperazine. And actually prochlorperazine, we have, as you can see, that number of molecules were suggested to us. We did some preliminary studies at our end. We did docking simulations also. Although all molecular docking and simulation data were given to us, I cannot present it here because it was all patentable data, but it was given to us by one of the Canadian lab, and one lab from IIT Delhi, and one from the CDAC Pune. And from that whole bunch of the molecule which were given to us, we have created our own preferences, and we could find 3-4 molecules, and one of them, prochlorperazine, which has given some good data to us, I am presenting today for you. Now, the question arises, what is prochlorperazine? So prochlorperazine is a I am showing you here as a radiosensitizer, but actually it is an antipsychotic drug. And there was some small study was already proposed by some of the Bach scientists long back, and they have shown its inclination as a radiosensitizer also. So there was some minor studies were reported, and so I have taken the lead from there also, and that gave us a little bit confidence also to work on the prochlorperazine. And while we were doing the prochlorperazine work, so as I told you that it is an antipsychotic drug, and so we started working on this molecule. And first of all, as I told you, that I have to find out that whether it is binding to the KRAS or not. So as all of us know that the KRAS mutation causes is one of the cause of the cancer, and it has been shown that almost 36% of the cancers are reported because of the KRAS mutation. So in the KRAS, the mutations occurs, most of the mutations which are occur in the cancers, and it is more common in the lung cancer, in some of the head and neck cancer, in some, and say prostate cancers, a couple of cancers it is reported. And so we have taken the most common one, and the one which we have taken was the KRAS G12D and KRAS G12C. We have worked with the KRAS G12S also, and other KRAS. We have worked actually with 6 mutant, but today I'll be showing you the data only with a few of them. And I have tried to make my presentation as short as possible. And this work has been recently published in FRBM. So if you want any detail, if you feel any problem in not understanding it because I'm going very fast, you can always say consult that paper, or you can write to me. I can explain you people accordingly. So when we try to look at the prochlorperazine binding, and this was the work done in my laboratory, so I'm only presenting that work which was done in my laboratory. So when we did with the KRAS G12D and KRAS G12C, the only important thing which I just want to communicate over here, that a very good finding we observed that when we did the KRAS, when we did the binding in 100 nanosecond simulation studies there, with the wild type, the prochlorperazine was not binding proper as strongly as it was binding to the mutant protein. So it was showing very good binding with the mutant proteins, which was a good start for us, and that gave us a confidence that it is binding strongly with the mutant one. So it means it is even able to differentiate between the wild type and the mutant. So this is all a typical RMSD plot, and it is showing you that it is interacting with particular amino acids. So those kind of details are given over there. And similar work we did, this was with the G12C mutated KRAS, and this is with the G12D mutated KRAS. And you can see over there, and we could find that when we did the same thing with the wild type, then actually from another one end of the loop of the KRAS, the prochlorperazine has got protruded out. So then what we did, and we have done the binding study with the GDP also and the GTP also. We have chosen the GTP bound conformation also and the GDP bound conformation also, in order to see that what kind of a difference they provide there. And this is with the G12S. We have done it because we were doing this work with the different cell lines, mutated cell lines, and those cell lines were having the typical mutations. Some of them were having G12S mutation, G12C mutation. So we were trying to do the modeling studies also, computational studies also, with all kind of the mutants. And then so the first experiment which we did, which is comparatively a new experiment now, in last 5-6 years, it has become very common. That is, you have to see that if any molecule is binding to any protein, it should be able to stabilize that protein at a certain temperature. So that is called as the SETSA, and we did the SETSA experiment, and we have seen that when the PCZ binds to the KRAS mutant cell line, we have PCZ is the short form of the prochlorperazine. So when it binds to the KRAS mutant cells, we could see that it was the protein was stable up to the higher temperature as compared to the unbound state. Similarly, we did, as all of us know, that if you want to show that any molecule is a radiosensitizer or a radioprotector, so the gold standard for any radiation biologist has to do is the clonogenicity assay. So we also did the clonogenic assay, and we have chosen some of the mutant cells there, A549, NCIH 460, NCIH 230. So we have chosen a number of KRAS mutant lung cancer cell lines, and then we have chosen some of the wild type cells also. And when we chosen the wild type and when we have chosen the mutant one, there was a very clear cut difference in the clonogenicity assay was there, and we could not find the RER was not crossing 1 in case of there was no radiosensitization was occurring in the wild type, whereas there was a very clear radiosensitization was occurring and the cell death was increasing in the mutant cell lines. So then what we did, that we have done some cell cycle studies, and we also did certain Western blot studies in order to see that how the PCZ is affecting the cell signaling in the cancer cells, and what are how this signal is passing and how it is actually affecting the cell signal, the checkpoint 1 and checkpoint 2 in the cell cycle. And while we were doing these experiments, we have looked into the NF-kappa B P65. That is, NF-kappa B is a pro-survival protein, IKK alpha beta, which is actually the regulator of the NF-kappa B, cyclin D1, CDK4, CDK6. And if you look into that you can see here that when the DR2 means when we have chosen the radiation only treatment, and when we have used the PCZ along with the DR2, you can see here that the expression of NF-kappa B was reduced. IKK alpha and IKK beta was also reduced, which was suggesting us that, and the CDK4 and CDK6 was upregulated, which clearly suggests that the pro-survival signal was going down, whereas the checkpoint is getting installed over there in the presence of the drug plus radiation. But when we did the same experiment in the non-mutant or the wild type cell line, the difference was, the results were completely different, or you can say reversed. Now, there was another experiment which we did, that prochlorperazine upregulates P53 and P21 expression in A549. So we wanted to see that how it is really affecting the P53, and because P53 is also one of the mutant which are found in most of the cancer cells, and the RAS and P53, RAS, RAF, P53 are correlated there, and they have a very important role in the radiation. So in order to see that, when we see that, we could find that it upregulates in A549 cells where the KRAS is mutated, the P53 and P21 expressions were upregulated, which was not found in the case of the H1299, which is not given over here. And then we did the typical DNA damage assay, and which is called as the gamma H2x assay. And when we did the gamma H2x assay, we could see that the phosphorylation of H2x protein was increasing in the presence of the PCZ along with the radiation as compared to the radiation only or PCZ only. So then we moved from our in vitro data to the in vivo data, and we have calculated on the basis of the human doses. We have calculated the mouse dose, because the human clinical dose is, because it is already a drug, so human clinical dose is very well defined for this molecule. So we have calculated the mouse dose from there, and then we started doing we made the xenograft model of number of cell lines. We have done some work with the head and neck cancer also, but I'm here presenting only on the A549 xenograph. And when we use this, we could very clearly see that it was actually radiosensitizing the xenograft, and it was, as you can see here, that it was increasing the survival of animals in which the radiation and PCZ both were given. This was all focal radiation was given. So tumor burden was reduced in the presence of PCZ and radiation as compared to the radiation only or the PCZ only. So then we did some, as all of us know, that the Ki67 is one of the measurement of the effect of the say cells. It is once again the marker of the cell proliferation, and you can see that the cell proliferation was substantially reduced in the presence of PCZ plus radiation. And then we have looked into some of the, I have removed these slides from here. We have done the proteomic analysis with this molecule, and there is a...
Sorry to interrupt you, ma'am. Please conclude.
Yeah, so we have done the RPPA analysis with this molecule, and we have found that it was actually PCZ in combination with radiation is altering the metabolic pathways. And I would not be able to explain it, but maybe sometimes later I would like to clarify it. And then we have looked into some other pathway, that is the ERK to ERK MEK pathways, and we could find that it was actually leading to it was modulating the RAS, RAF, MEK, ERK pathway, and it was leading to the apoptosis of the cell when the PCZ and radiation was being used together. So with all these things, I have just removed the whole data, and we have done a lot of other experiments, and we have tried to find out the total signaling pathway of the radiation of the prochlorperazine. And by what we have done, that we have made the permanent cell lines of the KRAS mutated cell lines, and we tried to do the experiment with the mutated cell lines, permanent cell lines, and we have shown very clearly there that by the immunoprecipitation and all other data, that KRAS, that prochlorperazine actually goes and binds to the KRAS. And we have done the KRAS inhibition assay also, which I have not included over here. So with all these things, the only summary which I would like to present here, that prochlorperazine can be a drug, can be a good candidate for the radiosensitization. And obviously it needs a lot of other studies to be done. And in 2021, we could publish it in the FRBM. We are still working there. And a lot of collaborations we have been doing, and we have got some money from, not on this project, from other project. We have from the ICMR and DBT. And this is the girl, Kirti, and the another girl here, Pragya, who have done this work. So I would like to thank them. This is one of my, 2020-21 group, which I just wanted to show you here. So with the hard work of these students, I could present this data. Thank you very much for giving me opportunity. Thanks.
Thank you, madam. Thank you for your nice lecture on prochlorperazine as radiosensitizer. It was very informative, interesting, and highly appreciable. Thank you, madam.
Thank you.
Any question, please? No. Next.
Yes, ma'am.
Fantastic work. And I remember this repurposing session in 2014 in Delhi meeting. We had one full day session.
Yes.
And as you mentioned, this work took off after that, and I'm glad to see such a lot of work has been added on. Yeah.
Yeah.
And in this interesting study, you had taken KRAS,
Yeah.
Yeah. See, I was just looking at some of your data. I mean, this is too specific a question. Maybe we can discuss later, but I still want to ask just one precise pinpointed question.
Yes,
When
yes,
you
sure,
are
sure.
talking about P53 impacting the transcription factor P21. Yeah?
Yeah. No,
Yeah.
what, so what do you mean to say that you are saying that P50, because of P53, P21 is getting affected? That's what you're saying.
I thought that is what you want to communicate. I just want clarification.
No, I didn't want to communicate that. I just wanted to communicate to you that it was impacting the P53 and P21.
So separately.
Yeah, so I'm not saying that they are affecting each other. And actually, when you remove the data without any, correlation, the things become very different. I tell you what happened. Actually, A549 cells are P53 mutant, but they, sorry, P53 null type. Fine. So when you are presenting this data, that data was not in the A549, that was actually in the different cells. Fine. But I just wanted to tell you one more thing here. I just wanted to communicate that somewhere this molecule was also affecting the P53 and P21. Which answer we have to still find out. And that answer I also don't have, but I purposely wanted to leave it there for you people, that maybe there is something occurring through P53 or P21, not through KRAS only. Because the reviewers were not ready to accept that anything can go and bind and replace GTP and bind more strongly than the GTP at the GTP binding site. So yes, you are very, very correct, and you have caught at the right place that the P53, P21 is also being, upregulated and down, you can say modulated in the cells. So what roles they are playing, that answer I have to find out.
Yes, ma'am. Actually, our redox biology paper on NRF2 keep one pathway.
Yeah.
It's basically a review by one of my students, and this is the point. At that time also, it was in our mind, and it's published somewhere in 2015 or so. And you can have a look at it. We have very clearly highlighted this point, that this connect is a missing in the study, and it is missing till date.
Yes. Yes, and I could not connect it, and we may discuss it on the telephone. Maybe if we sit together, we may connect it.
We may. Let the session move on. Thank you so much.
Thank you,
Dr. Vibhavan.
Rohit, hello.
Yes, sir.
Do I have the permission to ask a very short question?
Go ahead, sir.
Okay.
Dr. Vibha, nice presentation. I remember your presentation in Mangalore.
Yes, yes.
And it's an old memory which has come up. Dr. B.B. Singh's lab in BARC had done a lot of work in the 70s and 80s on
Holugal has done
drugs.
it. Yes.
In vivo, in vitro, a lot of work was done. We have summarized in some of our earlier reviews also, compared and all that. Now that it is something like, that's what I was going to say, that possibly something like, again, these drugs are being resurrected for potential cancer therapy adjuvants. Rather than getting into any discussion, I would just request you one thing. If you have some recent review on that, if you can send it over to me by email attachment, I'll go through it and I will understand the implications of your wonderful work.
Thank you, sir. I will send you. Thank you. Thanks a lot. Thank you.
Thank you, sir. Once again, thanks, Ma'am Dr. Vibhavan, for your informative and interesting lecture on prochlorperazine as radiosensitizer. Now I invite Dr. Damodar Gupta, INMAS, New Delhi. I would like to give my mic to ma'am. Please introduce you.
I would like to invite Dr. Damodar Gupta. He is from INMAS, New Delhi. He is delivering his lecture, invited lecture on TLR, economists, MOS in modification in biological radiation responses. Dr. Damodar Gupta.
Am I audible?
Very much, sir. Please start your talk.
Give me a second, madam, please. There is some technical issue.
Sir, be precise. Time is 15 minutes.
Right clicking.
Technical issue. I'm not able to share my slides on Mac.
Dr.
Then
Damodar,
other one
your
starts
share
first.
screen
Let
is
me resolve
green?
this problem.
Your
Maybe
share
the
screen is
speaker can
green?
come first. This technical problem I need to resolve. It says I have to change some system preferences in my Mac, and then I'll be able to present my slide.
स्क्रीन शेयर करें इसी तरह इसमें उसको जोड़ते हैं हाँ नहीं सर एग्जिट हो गए वापस ज्वाइन करेंगे
Dr. Gupta, is any problem? Now, I invite next speaker, Dr. Veena Kushwaha from DTU, Gorakhpur. And Dr. Veena Kushwaha. Dr. Veena Kushwaha.
Hello.
Yes, ma'am. Start your presentation.
मैडम मैं मेरे को स्लाइड शेयर करने दीजिए प्लीज.
Okay.
हेलो एक सेकेंड मैडम, लेट मी शेयर माय स्लाइड्स. एक्चुअली मैडम एकदम से मुझे बोल दिया गया तो स्लाइड शेयर करने में थोड़ा सा इशू आ रहा है. मैं बस एक सेकेंड में स्लाइड शेयर करती हूँ. शेयर स्क्रीन.
Dr. Utpal Ghosh, be ready after 15 to 20 minutes. We will be having your lecture.
Sure, sir. There were some technical issues in my.
I think my slides are visible.
Yes, ma'am. Go ahead.
Hello.
Be precise.
I hope my slides are visible.
Yes.
Okay. First of all, first of all, I would like to thank and yes, sir.
We are running out of time. Please be precise regarding the time. Thank you.
Okay. I'll try to shorten it more. So, congratulations to the organizing committee for organizing this long-awaited program and for inviting me. Thanks to Dr. Purohit and his team. I'll be speaking on impact of ionizing radiations on biomolecules. As all you people know that all the living organisms are built up of molecules. So, and that also of the biomolecules of same kind of monomeric subunits. The structure of a macromolecule determines the specific function. So whatever the changes are happening, whatever we are talking about, that is happening in the biomolecules only. So to be short, let us start with the next slide. Went very fast. All the living organisms, they are exposed to the radiations, and as many of us must have, many of the speakers must have told you that there are various types of, various sources of radiations, that is natural and man-made sources. They cause different effects on the human beings, or you can say on the living organisms, and the effect depends upon the type of radiation, the dosage of radiation, and the duration of radiation, besides the genetic makeup of the individuals. So ionizing radiations and non-ionizing radiations, there are two types. And right now we will be talking about the ionizing radiations, which are major types of our alpha particles, beta particles, X-rays and gamma rays. So these radiations, especially X-rays and gamma rays, they have a lot of penetration power, and they have ability to remove the tightly bound electrons from the atomic orbit. And because of this ability, they can convert the atoms into ions. These ions, they are rarely, or you can say into the radicals, which are actually highly unstable, and they react with all the molecules in the proximity. So in ionizing radiations in the aqueous environment of the cell, they result in water radiolysis, and they rapidly generate the reactive oxygen species, that is hydroxyl ions, ionized water, hydrated electrons, and secondary reactive oxygen species products like superoxides and hydrogen peroxides. These radicals further generate additional cell damaging molecules by starting a cascade of intracellular reactions. The figure here shows you that the ionizing radiations are generating potent intracellular oxidants like peroxide. superoxide hydroxyl radicals along with the reductants and hydrated electrons. The endogenous ROS system, the reactive oxygen system propagation that already occurs in your cell, like mitochondrial electron transport chain, that actually increases, and the endogenous system leads to the further increase in the ROS of the body. The reductive stress is also there, and that is the oxidative stress, and the reductive stress actually induced by the ionizing radiations leads to the loss of sulfur in the proteins, and especially in the amino acid residues of methionine and cysteine. So the cellular macromolecules are modified by direct ionization and via the reactivity of the high energy species originating from the water radiolysis. That is, water radiolysis we consider as indirect effect of the ionization. It results in indirect effect of the ionization, affecting an estimated 2,000 primary ionization events. So direct radiation damage is initiated with breaking of the bonds like SH bond, OH bond, NH bond, and CH bond. So the cells, basically there is a mitigation plan also with the cells for this IR dependent effects, and that is a very complex network of ROS metabolizing molecules and the detoxifying enzymes. And the enzymes are generally the oxidases and mutases, which help in detoxifying these ROS metabolizing molecules, ROS molecules. The determinantal effect of IR thus involve a series of events and is amplified inside the cells by endogenous molecules, and they basically damage the nucleic acids, lipids, proteins, and polysaccharides along with the other metabolites. This molecular damage caused by ionizing radiation in the cells also indicates that biomolecules are selectively modified. So to justify these two statements of mine, I'll be telling you how they interact with different molecules. Some interactions we will discuss and some of the biological consequences. And we have been developing the methodologies and techniques to estimate them so that they can be used as biomarkers of the infrared radiation effects in the therapeutics also and in the further technologies also. So DNA damaging events inflicted by ionizing radiations include the deleterious alteration in bases and sugars, cross-link formation, single and double strand breaks, and DNA clustering. Water radiolysis products like hydroxyl ions is the most abundant and particularly destructive to the nucleic acid molecules. Both nucleobases and deoxyribose are targeted by the hydroxyl ions and they mediate the damage. Radiation damage to deoxyribose is the primary event which underlies the strand breakage, and when the strands, and it is of the high frequency and it occurs randomly, and this results in the damage to the DNA backbone and the DNA backbone in response to both direct hydroxyl ion attack and as the activity of nucleic acid binding enzymes. That means that these hydroxyl ions, they attack the deoxyribose sugar, they actually damage the DNA backbone by attacking as well as by affecting the activity of the nucleic acid binding enzymes. Double-stranded breaks originate through the coordinated reactivity of two hydroxyl ions at nearby ribose sites. So ultimately it leads to the strand breakage through subsequent radical pathway. Both hydroxyl ions and the electrons in the aqueous environment react with the nucleobases, adding to unsaturated bonds and abstracting hydrogen ions from methyl and amino substitutes. So what happens is these radical products are structurally diverse and they are involved in many secondary reactions as oxidants or reductants depending upon the structure and reactivity of the species which is in the proximity. So what is happening actually in the nucleic acid, the radiations are inducing nucleobase lesions, and they also include the oxidative modification of the bases as well as the basic sites. But immediately the strand does not break. It starts breaking after all the lesions have come. For purine nucleobases, hydroxyl ions add at C4, C5, and C8, generating reactive adduct radicals that lead to the variety of products, with the most common being the 8-hydroxypurine, specifically 8-oxyguanine, which serves as a well-known hallmark, or you can say a biomarker for oxidative DNA damage. Pyrimidines, The olefins are also susceptible to hydroxyl ion addition, particularly at C5 and C6, generating pyrimidine glycose in the presence of oxygen. So aerobic conditions are required for that. Effect of ionizing radiations on cellular DNA oxidative damage to nucleobases and riboses mediated by hydroxyl ions is shown in this figure. The guanine is converted into 8-hydroxyguanine. Thymine gets converted into thymine glycol in the presence of hydroxyl ions, and we see major damage to the bases, damage to the strands. The strands they start breaking, and abasic sites are created. Now, what are the biological consequences? The immediate response to ionizing radiation induced oxygen species and reactive nitrogen species mediated DNA damage is activation of your cell cycle checkpoints. Thus, this cell cycle checkpoint response is activated. It is an intricately controlled network involving sensor, transducer, and effector proteins. So the response that basically respond to DNA damage signal, and they initiate a cytoprotective response. So that the DNA damage, this is actually a DNA damage response so that the damage can be controlled. So sensor and transducer proteins, they recognize the damaged sites and they start initiating and they start amplifying the biochemical cascade. The principal proteins implicated as sensors and transducers damage. They include MRE11, RAD50 complex. There are many checkpoint proteins which are included. But the activities of these proteins is tightly regulated by multiple modifications. Cellular DNA damage after ionizing radiation exposure is largely dependent on the capacity of cellular proteins to prevent and repair the DNA modification, which is induced by ionizing radiation. Now, how they react with lipids? Lipids are main components of your cell membrane, and they are estimated to be 5 nanometer thick. When they are exposed to these reactive oxygen species in the aqueous environment, what happens? They directly damage the lipids. Indirect damage induces, they directly damage, but that direct damage is of very less, it is not a large contributor. The large contributor is the indirect damage which is induced by water radiolysis to the product, right? So overall lipid modulation by the infra-ionizing radiations, the radiations they induce lipid peroxidation, particularly in the polyunsaturated fatty acids, and they lead to an increase in the membrane permeability, disruption of the ionic gradient, and transmembrane processes. And they also alter the activity of membrane-associated proteins. So induction of apoptosis is also observed in the when ionizing the cells are exposed to the ionizing radiations, and the targeted they target the cell membrane via increase in the ceramide level, and this is observed in the cells without a nucleus also. So this indicates that the ionizing radiations, they damage the cells independent of nucleic acid damage also. So they can damage through the lipid damage also. This is the slide which shows the effect of ionizing radiations on cellular lipids. Oxidative cis-trans isomerization of polyunsaturated fatty acids is shown. Reactive aldehydes are generated by the lipid peroxidation, and there are certain chemical probes which are used to study all these things. Unsaturated fatty acids, though they are unreactive to the molecular oxygen, but they rarely get oxidized by the radical mediated processes. So what happens is peroxidation of polyunsaturated fatty acids such as linoleic acid, arachidonic acid. This leads to the generation of hyperoxides, cis alkenes, and it leads to the degradation of the molecule into small carbonyl compounds or small fragments. These alkene in isomerization and peroxyl radical formation and the aldehyde production, they basically they form the radicals, and they persist in the cell for approximately 2 minutes. And these 2 minutes are the ample time for these radicals to modify the proteins, to modify the nucleic acid and other biomolecules, right? So what happens biologically? Moderation leads to of the lipid moderate level of lipid hyperoxides activates the oxidative stress response, and this excess when they are in excess, it leads to apoptosis. Then high levels and widespread level of lipid hyperoxides, They damage the cell membrane, the intracellular content, and they trigger the membrane lysis and necrosis. The sphingolipid ceramide, which was produced in the cells as a result of ionizing radiations, they basically hydrolyze and they generate a lot of ceramide, and this ceramide from the sphingomyelin within the membrane forms lipid rafts. And these lipid rafts alter the membrane properties largely because ceramide contains lipid rafts and they form a ceramide enriched membrane platforms. Now, lipid platforms, they not.
Veena B. Kushwaha, ma'am, is it audible to you, ma'am? Madam, is it audible to you?
Unmute. वो बोलती जा रही है वहां से रिकॉर्डेड नहीं होगा नहीं स्लाइड चेंज रियल टाइम लाइव.
Sir, you are audible. Only thing is, I think our voice is not reaching to madam.
Yeah.
It seems she is presenting using her mobile. Maybe.
Hello.
Yes, ma'am. Yes, ma'am. Ma'am, we are requesting.
Yes, sir. What's happening?
Nothing, ma'am. We were requesting to you just to sum up the presentation, please.
Hello.
Yes, ma'am, please.
Hello. Should I continue?
Yes, please continue and sum up within 2 minutes.
Okay. Okay.
Yes, yes. Thank you. Ma'am, please unmute yourself.
She was audible earlier. Now we are not able to hear her.
Sir, am I audible now?
Yes, madam, you are.
Now you are audible.
Okay. Let me conclude then. I have actually stopped sharing my slides. So let me conclude by saying that the determinantal effect of ionizing radiations involve highly orchestrated series of events that are amplified by the endogenous signaling and culminating in the oxidative damage to the DNA, lipids, proteins, and many metabolites. So despite the global impact of ionizing radiations, the molecular mechanism underlying the tissue damage suggests or reveal that many molecules are chemoselectively modified by the ionizing radiations. So there is a lot of development going on in the technologies, especially in omics for mapping DNA and protein modification, which can revolutionize the study of ionizing radiation effect on the biological systems. So we need to study in the cell tissue biological fluids, and we need to identify the molecular features, biomarkers for the ionizing radiations exposed and responses. For this work, basic things should be known to us. A combination of omics technology like genomics, epigenomics, proteomics, and metabolics can be is anticipated to lead to the new hypothesis in understanding the ionizing radiation effects. And I think with this, I would like to stop here and to discuss with all of you in details whenever next time I get a chance. Thank you.
Thank you, ma'am.
And I'm sorry I was not able to hear the warning in between.
Thank you once again. It's our pleasure to hear you. And you delivered very informative and very nice talk. Thank you, ma'am. Any question?
If I may ask, just very briefly.
Yeah.
Very brief. What kind of work is being carried out in your lab, Dr. Veena?
Sir, I actually work with phytochemicals, and we try to mitigate the oxidative stress which is produced by the chemicals as well as radiations. Radiation, we are doing very less sort of work.
So, but here there was there any data you gave?
No, I haven't given any data, sir. But if you are interested, I can mail you, or we can talk afterwards. This was actually a popular lecture type of thing, whatever we observe in our normal studies.
Okay. Anyway, then it was going 45 years back, a good refresher course for radiation biology.
Maybe, sir. Actually, I spoke for students.
Okay, ma'am, thank you. Now I invite Dr. Damodar Gupta in Mass, New Delhi. Dr. Damodar Gupta.
Thank you so much, madam. I hope I am audible now.
Yes, yes, please proceed.
Is my slide visible?
Excuse me, Dr. Gupta, please be precise regarding the time because we are very much out of time.
So I'll try to finish in 10 minutes, sir. Not
Thank
more
you,
than
sir.
that.
So very kind of you. If you will do
Yes, sir.
you will be very much
Sincere apologies for technical glitch that has happened earlier. And I would like to thank organizers for inviting me to share research work that we are doing it in mass. And also Society President Dr. Madhubala, who is well aware about this particular work that has been going on in our institute.
So please leave all these formalities and start your presentation.
Okay. So the topic of my presentation is TLR agonist, menon oligosaccharide in modification of biological radiation responses. As you know, the TLR agonists, toll-like receptors, they are very much required for our immune system that can recognize different kind of patterns, and they are available on cell surface. And they recognizes not only foreign particle or foreign molecules which comes to our body either from the pathogen, they can also recognize DNA, RNA, or inflammatory response that is being generated in our body. So whatever has happened during the COVID eventualities also, either in septicemia or other things, these toll-like receptor, they played very big role in relation to inflammation or in relation to protection of that particular individual also. So there are different toll-like receptors that interacts with different kind of biomolecules like TLR5 that interacts with the flagellin, TLR3 double-stranded RNA, TLR2 they interacts with different kind of molecule that comes from fungus or other things. And as soon as they interacts with various biomolecules, they initiate intracellular signaling in such a way that cells can help in survival of that organism. So this kind of signaling or this kind of approach can be taken for achieving radiation protection. So screening or identifying those toll-like receptor agonists are very much important in relation to achieving radiation protection, so that they can activate either NF-kappa B pathways or other pathways that can alter mitochondrial physiology. My job is made very easy by the previous speaker who has shared so much about the free radical generation and their reaction with the various biomolecules and how it affects our cellular physiology. As you all know that free radical, it is generated by low linear energy transfer radiation that can interact with the various biomolecules. And if we can control somehow this radical generation, we have better chances of achieving radiation protection, specifically against gamma radiation or X radiation, low linear energy transfer radiation. So our approach was to identify such a molecule that can alter signaling pathway that is related to NF-kappa B, that can alter mitochondrial physiology, which is very much related to the generation, or it is major site of oxidative stress within our body, a molecule that can protect GI tract as well as hematopoietic system along with boosting the immunity. So toll-like receptor, this receptor has very good tendency so that they can create intracellular signaling pathway in such a way that every single thing can be regulated very easily, along with the synthesis of cytokines which are required for the radiation protection. So identifying this molecule, we have to identify or develop some kind of biological sensor so that we can screen all these toll-like receptor agonists along with various phytochemicals or chemicals which are available in the market, already in use for various treatments. And we have developed various different kind of cell-based biological sensors. This has also been utilized for radiation biodosimetry purposes also. That will be shared by Dr. Shweta Sanguri in coming presentation. I think after me or later on. So this molecule that we have identified, it is menon oligosaccharide, which is already well-known, available molecule as IMOS in the market. It is chewable tablet, which has been taken as a prebiotic. It is also being used as a adjuvant. So all this data is available in public domain. What we have done in our institute is identifying its potential as a radioprotector, and we know how it is protecting by activating NF-kappa B pathway, by activating altering mitochondrial physiology. Its molecular target is TLR4, and it is protecting through GI tract as soon as we take it orally. Now, how to assess radiation protection? It is mainly for the student. This slide, the mitochondria is major site of oxidative stress. Not only oxidative stress, energy is also being produced in mitochondria, which is very much required for repair and recovery of that individual organism. How it is being generated? Through electron transport chain system. If somehow we can control this free radical generation or electron transport chain by altering or by reducing or by taking cells to the hypometabolic state, we can really achieve radiation protection and reduce the damage by reducing the damage to the various biological molecules. So first aim was to identify whether menon oligosaccharide can really activate NF-kappa B pathway or not. The developed cells or a reporter biological sensor that was developed in our laboratory has clearly shown that yes, menon can really activate NF-kappa B. Now next aim was to identify whether it can really alter mitochondrial physiology or not. If you can see this histogram or dot plot in different gate, near about 47% population is showing active population. As soon as we add dihydroxycarbocyanine iodide, DIOC6 dye, it gets active cells or active mitochondria can take this dye and show major uptake. Within 10 minutes, everything gets saturated. So if we observe near about 47% of this dye, 47% of population has a good amount of dye. With respect to the other gating population which was treated with the menon oligosaccharide. So we have identified that yes, at one particular time that mitochondrial membrane potential is really suppressed. If we can irradiate our cells at this moment, when mitochondrial activity is lower, we may have better chances of getting radiation protection. So we have identified one particular window near about 1 hour or 30 minutes in which we can really expose our cells, these normal cells, and then get radiation protection. After 4 hours, this effect is completely gone. So we have this time window, 1 hour or 30 minutes to 1.5 hour time window in which we can get radiation protection in best possible way. Now, whether free radical generation is also in similar way or not, yes, it is. At this time, after 30 minutes or 1 hour treatment, free radical generation was also lower. So we have better chances of radiation protection. Yes, whether mitochondria is really playing major role or not, that we need to assess in further experiment. But after treating cells with the menon oligosaccharide, either it is 5 microgram or 10 microgram or 20 microgram, all these cells have shown radiation protection against 3-gray gamma radiation. We have identified 3-gray gamma radiation near about 37% survival or 40% survival. Now, whether mitochondria was really involved or not, we have treated our cells with the mitochondrial electron transport chain inhibitor. It was rotenone or antimycin A. And we have found, yes, mitochondria and electron transport chain plays major role in giving radiation protection in by the menon oligosaccharide. But these electron transport chain inhibitor were very toxic. So we had to develop another cell type, that is Rho zero cell line. It was Rho zero cells are completely mitochondrial electron transport chain deficient because they don't express or they don't possess mitochondrial DNA. They look little bit elongated, but yes,
Please conclude your presentation.
Okay, madam. So I'm finishing in 1 minute. Assessment of radiation protection and role of mitochondria was found to be measured in relation to TLR agonist mouse. And we have identified various expression of various mitochondrial electron transport chain complexes also. And it has shown, yes, mitochondria plays major role in radiation protection. And this is how what the mechanism and where we are at present. We have identified radiation protection near about 50% even in 12 to 13 month old animals also. This is first kind of study where we have used oldest possible animal for radiation protection studies, because we have to assess our molecule. It should provide radiation protection to all age group. And as you know that mitochondria, mitochondrial changes are there with respect to age, and there is decrease in radiation protection even in 12 to 13-month-old animal also. That indicates, yes, it provides radiation protection through mitochondrial electron transport chain pathways. And here we are. All this data is published. For radiation, this is for students.
मैं normally इनको separate रखता हूँ. चलो ठीक.
For radiation protection, if we know our need, that for what purpose it is supposed to be used, where it is supposed to be used, and whether it is clinically required or not. In case of radiation therapy, where we want to really protect normal cell instead of protecting transformed cells. So this molecule specifically protects normal cell, all the organisms, as well as survival advantage. We know its molecular target also. And thank you so much for giving opportunity for presenting my work. I know it was very short time, 10 minutes or 15 minutes is not supposed to be given.
We agree with you, sir.
But thank you, sir.
But thank you, Dr. Gupta, for a very informative and nice lecture on impact of ionizing radiation. Any question, please, in very short.
I have just a comment. See, some of these, can I make a comment?
Yes, ma'am, please.
Go
ahead. See, some of these presentations we have not been able to do justice because of the shortage of time, and they are from senior professors, senior faculty. So my suggestion is that organizers should request their published paper, because most of the work is published. Okay, and from this work, these published papers can be displayed on the ISRB website or ICRB website, so that people can understand this work.
Totally agree. Totally agree.
Yeah. So this is the so the published work can be displayed on the ISRB website or ICRB website, but we will not take the responsibility of breach of your agreement with your publisher. So you have to either summarize and give it to us.
I think it is already available in public domain. So most of this work, if it is in open access journal, it can be accessed and kept on our site also. It is really good idea, madam, to have such kind of data on our website or web page. That will be helpful for all the students. Thank you so much, ma'am.
Dr. Damodar, thank you very much for your nice presentation. I'm actually very sorry we are running short of time, and I agree with Dr. Madhuvalan also. It's not possible to conclude within 15 minutes. Dr. Damodar is a very senior scientist. I assure you, when we publish our proceedings, we'll publish the full paper. We would like to read the full paper also. Proceeding in some judice about the
Professor Vibha Tandon also.
Yeah.
And some other senior faculty members who have summarized their work of many years in small time.
No problem. We will publish full paper in the proceedings.
Give an open email.
With the Professor Madam Madhubala, we publish full paper. Thank you, Dr. Damodar. Thank you very much.
Thank you so much, sir. Thank you.
Hello. Now I invite Dr. Utpal Ghosh, University of Kalyani.
Yeah. Am I audible? Hello.
Yes, you are audible.
Okay, let me share my things.
Excuse me, please. May I request Dr. Sweta, be prepared for your lecture after 15 minutes. And Dr. Utpal, please conclude your lecture within 15 minutes. Thank you.
Yeah, I'll try my best to conclude it. Is the slide is visible? Hello.
Yes.
We don't know whether it is moving. So please put it on the PPT mode, full screen mode. And try to move it.
This is F5, yeah.
Yeah, now go on to the next. Yes, it's
Yes.
fine. It's fine, please go ahead.
Yeah. So thank you. Good evening, everybody. Thank you for organizers for giving me opportunity to share my work and thoughts over this conference. So this is the title of my talk. I don't know how do I accommodate all these things within 10 to 15 minutes. So I'll try my best. This talk, the title of this talk is High LET Carbon Ion Radiotherapy Could Be a Promising Approach to Cancer Therapy Against Highly Aggressive Human Lung Cancer Cells. So I will not go to the introduction part, and I have organized my talk in these 4 chapters, but I will skip this. And I will directly go to, but let me say a few words about the problem faced in the photon therapy. The radiotherapy could be by 2 ways, using photons, X-rays, gamma rays, or using particles, high energy particles, protons or carbon ions. And judiciously chosen chemotherapeutic drug could be combined with radiotherapy. So chemoradiotherapy is coming up and is giving a better effect than the single mode of treatment. Okay, there, if you have a tumor inside the body, then photon therapy is not the suitable one, because if you look into the energy deposition pattern of photons, you see here, with the depth, the energy decreases. And if you have a tumor inside the body, so maximum energy is deposited in the on the healthy cells, but less energy is targeted to the tumor cells. So on the contrary, if you use high LET particle like protons or carbon ions, they have a unique property of energy deposition pattern. It can deposit its maximum energy at the end of its particle track. This is called the Bragg's peak, and you can calculate the depth of the tumor, and accordingly the energy can be regulated, and so the Bragg's peak can be targeted to the tumor cells. So the normal cells are least affected. And there are other problems in the photon therapy, like it grows radio resistance and its ionizing density is lower than the particle radiation. So so actually particle therapy, they have certain advantages over the photon therapy. That's why this is becoming very promising. And since the physical nature of photon and physical nature of the particles are different, so when you expose the cells with a particle, the cellular response should be different, and it is different. And that's why the basic understanding of cellular response against particle therapy is utmost important to understand the rationale of using carbon ion or high LET particles in particle therapy. So, so I'm skipping this. So the most advantageous part of this carbon ion radiotherapy is it's very effective against hypoxic tumor, which is difficult to treat, chemo-resistant, radio-resistant tumors. And surprisingly, this carbon ion radiation, it reduces metastasis. I will show few of our works, the mechanism of reduction of metastasis. So here we have 3 objectives: comparison of DNA repair ability and radio sensitivity after gamma and carbon ion. So here we have compared these two type of radiations, and then I will show few slides about the mechanism of induction of apoptosis by carbon ion, and then I will show few slides where we have compared the cell invasive property and EMT pathway, epithelial mesenchymal transition pathway, in p53 wild type and p53 mutant cells. So I put it here, these 2 slides, because as you know that the almost 50% cancer cells are p53 mutant, and they are difficult to treat. And here I will show that how carbon ion affect these 2 cells. So you see here, the killing effect is more for carbon ion compared with the gamma rays. I'm not going into the details. And if you combine this carbon ion with the knocking down of a particular gene, PARP1, or you can use the inhibitor of PARP1, if you can combine this, it gives a very synergistic effect, and that killing effect is very, very high. So this data tells that judiciously chosen, as I said, that judiciously chosen chemotherapeutic drug can be combined with radiotherapy to get better effect. And here I will show you a few data where PARP inhibitors combined with carbon therapy, carbon ion exposure gives better effect. So we have also seen this carbon ion effectiveness over the X-rays and proton in different cell lines, chondrosarcoma cells. Here are the pathway of induction of apoptosis by carbon ion. I am showing very shortly. After carbon ion exposure, it triggers the fall of mitochondrial membrane potential that triggers the caspase 9 activation, followed by caspase 3 apoptotic body formation and DNA ladder. But surprisingly, we have observed that it directly activates caspase 8, which is generally activated by Fas ligand binding with the Fas receptor in the membrane. But how this carbon ion directly triggers caspase 8 activity is really not known. So we need to go in detail. Similarly, AIF translocation is also triggered after carbon ion radiation, and a reason is still unclear. So these are the whole pathway. I am not showing all the slides. So we did fluorometric assay, immunofluorescence for checking this AIF translocation, caspase independent pathway this is, and caspase is activation of caspase is using fluorometric determination as well as immunofluorescence assays. So so our third objective is comparison of cell invasive property. As I said that carbon ion irradiation, it reduces metastasis, and metastasis is the cause of most cases, the cause of the death of cancer patients. So so understanding the mechanism of reduction of metastasis is very important here, so that we can understand the rationale of using carbon ion in radiotherapy, or combining suitable protein inhibitor with the carbon ion. So here we have investigated matrix metalloproteases, MMP2, MMP3, MMP9, and its tissue inhibitor, TIMP1, 2, 3, their expression and their activities. So let me describe this slide is very important data. As I said that P53 mutant cancer cells are difficult to treat. Here we have shown that this P53 mutant cells, H1299 P53 null cell, is to some extent resistant compared to the P53 wild type cell to the carbon ion exposure. But if we combine this carbon ion with PARP inhibitor, olaparib is in clinical trial and showing very good result against several types of cancers. And when we combine this olaparib with this carbon ion, you see here it's sensitized. So it indicates that combining olaparib with carbon ion could be a good way to treat P53 mutant cells. Here are the comparison of wild type and mutant cells. So both the cases we observed the reduction of MMP's activity, but extent are different and pathways are also investigated here. These are the pathways. Actually, these signaling pathways, EGFR, AKT, P38, and ERK, they are the upstream event of activation of NF-kappa beta, which actually is the transcription factor of matrix metalloproteases. And we observed that carbon ion radiation drastically reduces MMP's activity, and that is through this pathway, EGFR, AKT, and P38, this pathway. And if you look in detail, so I don't have any time, if you look in detail, the pathway is pathway is active in both the cell line, if P53 wild type and P53 mutant, but the extent is different. So we need to check further details. But the take-home message is when we combine olaparib is a PARP inhibitor. When you combine olaparib with carbon ion, it gives better effect than the single mode of treatment. And this is the whole pathway we observed. And it is to be noted that the mechanism of reduction of metastasis by carbon ion is just opposite to when you irradiate the cells with gamma radiation, because gamma radiation, there are lots of reports, and we have also shown that gamma radiation triggers NF-kappa beta. But here carbon ion exposure reduces NF-kappa beta, this pathway. And we have a paper that where we have showed that olaparib pretreatment protects gamma induced NF-kappa beta activation. So in that case, that olaparib could be used as the protective measure. So I have not included that data here. So major challenges are that we have to radiosensitize selectively the cancer cells and radio protect the normal counterpart. So that is our aim, and for that we need to understand the basic understanding of cellular defense mechanism, cellular response against this high LET particle, as well as to understand the molecular mechanism of different cellular responses, so that we can judiciously choose protein inhibitor which can be combined with the carbon ion. And one the major challenges of carbon ion radiotherapy at a glance is that the infrastructural facilities for carbon ion therapy is very expensive, and in India still it is not established. But Tata Memorial Center is supposed to initiate this hadron therapy very soon. But in private sector, Apollo, they have started proton therapy. So anyway, these are the challenges. And so our conclusion is ionizing radiation breaks DNA and hence it interferes the DNA repair system, thus making it the radiosensitive. And carbon ion is always better killing effect than gamma radiation or X-rays. And it triggers the caspase dependent, caspase independent pathways, but the mechanism is still unclear. And carbon ion, it inhibits metastasis through EGFR, AKT, P38 and ERK signaling pathway. And when we combine this carbon ion with the rucaparib or olaparib, those are the inhibitors of polydipyribose polymerase, and they are in the clinical trial. Then we can selectively kill the p53 mutant cancer cells. So I would like to thank the IUAC, Inter-University Accelerator Center, where I have been using the infrastructural facilities for carbon ion beam. I would like to thank also the funding agencies, CSIR, DST, DVT, for their funding. And these are my research scholars and collaborators. Thank you very much for your attention.
Thank you very much, sir. Thank you very much, sir, Dr. Palghosh, for your very much informative lecture on high-lead carbon ion radiotherapy. May I have some questions, please, if any? So thank you, sir, once again.
Thank you.
Now I invite Dr. Sweta. Dr. Sweta, can you hear me?
Yes, sir, I can. Am I audible?
Yes, please.
Please allow screen sharing.
Madam, please listen. We are running out of time very much. So please conclude your lecture or presentation within 10 minutes. Thank you.
Thank you. I hope my screen is visible right now, is it?
Yes, go ahead.
Thank you very much. Good evening, everyone. My name is Shweta Sanguri, and I'm working as a research associate in IUAC New Delhi. It's an honor to be able to present my talk here. The talk is entitled Cellular Response to High-Lead Radiation, with special reference to mitochondria. A very brief introduction about biological effects of radiation. Ionizing radiation has sufficient energy to affect atoms in living cell and thereby damage their genetic material. If radiation interacts with atom of DNA molecule or some other cellular component critical to the survival of cell, it is referred to as direct effect. Such an interaction may affect ability of the cells to reproduce and thus survive. Ionizing radiation can ionize molecule or break chemical bonds, which damage the molecule and cause malformation in the cell process. It can also create reactive oxygen and nitrogen species that damage biological process and disrupt physiological process, including gene expression and protein modification. So based on the extent of damage of the cell may choose to undergo cell death or repair. So at organism level, very high level of radiation exposure delivered over a short period of time can cause symptoms such as nausea and vomiting, and sometimes result in death over the following days or weeks. This is known as acute radiation syndrome, commonly known as radiation sickness. Exposure to low levels of radiation does not cause immediate health effects, but can cause a small increase in the risk of cancer over a lifetime. So now exposure of cells to radiation adversely affects cells, and DNA is a critical target, and DNA damage by ionizing radiation has been extensively studied. Mitochondria, on the other hand, being the powerhouse of the cells, are one of the most important targets of radiation because they are the site of oxidative phosphorylation, where energy production occurs by utilizing molecular oxygen, which further increases the oxidative stress. Now, the emerging experimental evidence from research recent years have accelerated research in understanding mitochondria as an important target for ionizing radiation and its significance in cellular radiation response. So mitochondrial functions, which are closely related to integrity of mitochondrial DNA, play a key role in many cellular processes. mitochondrial dynamics is also vital for structural integrity and function of normal cellular function. Now, the cell lines which are used in this study are normal cells. So we have utilized normal kidney embryonic cell NK cells, and also reporter cells, human embryonic kidney cells, HEK293T, stably expressing subunit 9 EGFP and MFN2 EYFP proteins. Carbon ion irradiation was done using 15 UD pelletron in IUAC New Delhi. I have a short 40 seconds video which I would like to show at the end of the presentation if the time permits. Now, development of genetically modified cell-based reporter system. These are the reporter constructs that have been used to develop model system, but we have utilized two model systems out of these in the present work. One is MFN2 EYFP, another one is subunit 9 EGFP. Genetic manipulation and cloning. DNA cloning is a process of making multiple identical copies of a particular piece of DNA. In a typical DNA cloning procedure, a gene of interest along with the reporter gene and a selection marker is first inserted into a circular piece of DNA called plasmid. Next, the recombinant DNA is introduced into bacteria. Bacteria carrying the plasmid are selected and grown up. As they reproduce, they replicate the plasmid and pass it on to the offspring, making copies of DNA it contains. Now, development of genetically modified cell-based reporters. This is the next step. There are a wide range of transfection methods that include physical, chemical, and biological techniques. We have used retroviral and liposuction in our experiments. So the transfected cells are selected using appropriate antibiotic, and a single cell which is stably transfected is picked up from the pool, and the clonal expansion is done. These cells are then cryopreserved for future experiments. Now, a brief idea of what mitochondrial fusion fission homeostasis is and why it is important. So dynamic changes in mitochondrial morphology are basis of mitochondrial function. Mitochondrial dynamics involves continuous fission and fusion, forming a dynamic network to maintain their abundance, quality, and cell function. We have studied expression of important fusion and fission related proteins after exposure to carbon ion beam exposure at different time points, but I'm not showing the results here because we do not have very much time. MFN2 is an important protein required in fusion process of mitochondria, and these are the results after exposure to this high LET radiation. These results illustrate decrease in mean fluorescence associated with MFN2 protein 12 hours post carbon ion beam exposure in comparison to the sham irradiated control cells. We have obtained similar results from flow cytometry experiments and protein expression studies. Now, decrease in MFN2 associated fluorescence signify shift towards mitochondrial fission and consequently more number of mitochondria per cell. So this may be a strategy to increase a mitochondrial number to cope up with the energy demand to have more electron transport chain machinery and maintain bioenergetic status inside the cells. Now, in human, the OX4 system comprises 5 multimeric protein complexes which are present in inner mitochondrial membrane. And subunit 9 is a component of complex 5 of electron transport chain. So subunit 9 associated fluorescence shows expression of subunit 9 protein of complex 5. Now, radiation exposure to cells shows dose-dependent increase in subunit 9 associated mean fluorescence 12 hours post-irradiation with respect to the control cells, suggesting increase in expression of subunit 9 protein and in turn increase in number of electron transport chain machinery. Now, the compromised mitochondrial electron transport chain function may lead to abnormal mitochondrial morphology, like more fragmented mitochondria and mitochondrial dysfunction, like less ATP generation, increased mitochondrial oxidative stress. So the results show an initial decrease in mitochondrial fusion and shift of dynamics of equilibrium towards mitochondrial fission for first few hours. I'm not showing the entire time kinetics study of both these reporter cells. Here, the expression of protein involved in both the mitochondrial fission and fusion and mitochondrial electron transport chain was also investigated. And the protein expression profile corroborates with the current finding. Now, these are some of the results which were done with the normal kidney embryonic cells, normal kidney cells, which are NK cell epithelial. Normal mitochondrial membrane potential is a prerequisite for maintaining mitochondrial oxidative phosphorylation and ATP protection. So there was a significant decrease in mitochondrial membrane potential in NK cells 24 hours post carbon beam exposure, possibly due to damage and the uncoupling of electron transport chain. The result indicates decrease in membrane potential and consequently in ATP levels 24 hours and 48 hours post beam exposure. And the beam exposure created a major energy crisis as reflected by low cellular ATP content. There was overall increase in total and mitochondrial reactive oxygen and nitrogen species in cells and after exposure to radiation. Exposure to radiation increased mitochondrial mass 2 to 3 folds in NK cells in comparison to control cells. MTT assay is dependent on mitochondrial respiration and indirectly serves to assess the cellular energy capacity of a cell. The amount of pharmacin formed were normalized with corresponding cell numbers. Irradiated cells show multifold increase in amount of pharmacin per cell. This observation is substantiated by microscopic images of the pharmacin in control and irradiated cells shown on the right. Irradiated cells show very high pharmacin deposition per cell than control cells. To summarize, carbon exposure creates a major energy crisis in normal cell reflected by low ATP yield and alteration in bioenergetic related subunit 9 protein. It also reduced mitofusion to a level, which is mitochondrial membrane protein that participates in mitochondrial fusion in mammalian cells. Decrease in cellular ATP and MMP level signal cells to increase electron transport chain function to cope up with all the energy demands and also maintain mitochondrial membrane potential levels as indicated by increased expression of subunit 9 protein. Alteration in expression of protein involved in maintenance of mitochondrial dynamics and electron transport chain function affects physiological and metabolic states of cell. And high ROS levels and oxidative stress generally lead to abnormal mitochondrial dynamics, especially excessive mitochondrial membrane fission. So we have a dedicated radiation biology beamline, which is equipped with a radiation system, which is known as Aspire. This is a computer-controlled system that enables irradiation of cells with a predetermined dose. The cells are grown in 35 mm Petri culture dishes, which are kept in sterile media, and cells can be irradiated one after another at quick succession. I feel immensely privileged to be able to work in the only institute in our country that has in vitro cell culture facility to carry out high LET radiation work. This is a short video, almost getting completed. Thank you. I want to thank my supervisor, Dr. Asiti Kanta Sharma, not only for his teachings in field of radiation biology and radiation physics, but also his invaluable wisdom about life. Also want to thank my PhD supervisor and mentor, Dr. Damodar Gupta. Thank you, SRB and RK Purohit sir, for this opportunity today. Thank you very much.
Thank you, ma'am.
Shweta,
yes,
do you also have, may I ask a question, ma'am?
Yes, of course.
Shweta, do you also have a method to do the dosimetry, the dose absorbed?
Yes, ma'am. Actually, we have also tried to utilize these reporter cells as radiation biodosimeter, and we have also published a paper regarding that. We have done it with gamma radiation, still on the process in developing it with high LET. So
No, cross check. I mean, how do you actually verify how much dose has been given?
Okay, so ma'am, we already calculate the dose, what dose we are giving to the cells, and then we either utilized flow cytometry. Usually flow cytometry, we have done few experiments with flow cytometry, and then we record the fluorescence, the change in fluorescence pattern with increasing radiation or decreasing radiation dose. And also the different time points after radiation also play a major role. So we have tried to derive a relationship.
So this is all qualitative. Absolute dose absorption is not known, or at least delivery.
Ma'am, we have derived a relationship, which is like, I would not say that it will be very accurate to maybe 0.5 gray, but then yes, we have a relationship. We can actually try and relate to the fluorescence level and then derive how much radiation dose was absorbed by the cells.
Oh,
Shweta, ma'am's question is about the dosimetry, how you do the dosimetry part. I think you use carbon-12 electron beam.
Oh, I thought.
So 85 MeV. So how much dose is really absorbed by the cells? So with the pulse, it is supposed to be done. I think her question is that one, if I'm not
Yeah,
wrong.
there is a way to, because this Asiti's facility has been coming up, it's been evolving. Since 1988, I have been knowing, and we've been working together in IUAC. And the biggest challenge was in the high LET radiation is how to regulate the dose which is to be given, and if we can actually quantify the dose.
Oh, ma'am, it is actually, I'm so sorry, I did not understood your question correctly. Yes, we have a method. We do use a formula wherein we can directly convert the number of particles to the gray, to the doses in gray. So like we calculate it and then we deliver the precise dose. It is possible.
Okay,
yeah.
Thank you.
I'm sorry, I misunderstood the question.
No problem.
Thank you, ma'am. We are very grateful to all the invited speakers of this session. All the talks were presented in excellent manner. Speakers put their special efforts in preparing their presentation. Thank you very much once again. Now over to Dr. Aruna Chakravarti for report of this session.
Thank you, ma'am. ICRP 2020, organized under the auspices of Indian Society for Radiation Biology, radiobiology in the coming decades, January 19 to 21, 2022. Today is second day in session 6. Total 5 invited presentations have done. All presentations or invited talk were very nice, interesting, knowledgeable, and session was very informative. Thank you all.
Thanks all the invited speakers of the today's last session, 6th radiation cancer. On behalf of ISRB, on behalf of ICRB Organizing Committee, from the core of my heart, I thank Professor Vibha Tandon, Dr. Damodar Gupta, Madam Veena, Utpal Ghosh, Shweta ma'am, and of course, our Honorable President, Dr. Madhubala ji, and Professor Vijay Kalia ji, and the audience still in the sitting in this auditorium. I would request now Dr. Anila Purohit, Dr. Pratap Singh, Dr. Sonu Shiva, you all come on the dais and present memento and certificate to our honorable chairpersons. First of all, I would request ma'am also, Mrs. Pratap Singh, ma'am, you also come on the dais. Dr. Manisha, Suresh sir, you also come on the dais. Suresh sir, Pooja, you also come on the dais. First of all, we present the bouquet and memento to Dr. Dipti Srivastava.
First of all, Dr. Pratap Singh ji, come here. Pooja, you also come here. Certificate to the chairperson, Dr. Dipti Srivastava, senior faculty members of Department of Zoology. Since 9:30, she is with us. So yes, sir. Pooja, you come. For a while, you move the mask. May I request Dr. Anila and Dr. Sonu to present memento, our honorable chairperson, senior professor of Chemistry, Dr. Jaishankar Acharya. Sir, we are thankful to you for sparing your valuable time for this interesting conference. Dr. Soniswa, Dr. Leela, Dr. Pratap Singh, Dr. Manisha, Aruna, Suresh sir. Thank you so much. I will look at Dr. Suresh Sharma sir. You will give memento to Dr. Aruna Chakrathi, Organizing Secretary of the Industry Conference. Please go. Yeah, Aruna Chakrathi, Dr. Neela, Dipti ma'am. Please. Sonu ma'am. Sonu Anila ji, Sonu madam, I also. Yeah. We are especially thankful to Dr. Anil and Dr. Sonu Siva for since morning 9:30 you are with us. Thank you so much. Thanks Dr. Anna for chairing this session. Thank you all. Tomorrow we all will meet tomorrow in the morning. We will share the latest schedule. Thank you so much. Thank you, ma'am.